Hello there, and welcome to Life Beyond Seen, where curiosity settles in gently and science takes its time, the way a tide rests against the shore before it pulls away again. Tonight we're drifting somewhere different, not into the places where the ocean roars or dazzles, but into the places where it barely breathes at all. The world under the waves is vast and loud in its own way, full of colour, motion and endless change. But there are corners of it where life hangs on by the thinnest thread. These are the quiet regions, the fading areas where the water loses its oxygen and the brightness of the sea dims into something still, heavy and waiting. They're called dead zones, though the name doesn't tell the whole truth. Even in the places where oxygen slips away and the currents slow down, something manages to endure. Something always finds a way to keep going. As we settle into this topic, imagine yourself drifting somewhere calm, not as a diver or a swimmer, just as an observer hovering above an ocean that looks perfectly ordinary from the surface. You wouldn't know that, beneath the soft rise and fall of the waves, there are areas where fish can't survive, where most creatures avoid passing through for more than a moment, where only the most stubborn or resourceful forms of life remain. We're heading there tonight, taking our time, letting each detail arrive naturally, because the story of what survives in these low oxygen pockets isn't rushed. It's steady, patient, and stronger than it looks, just like the organisms that live there. Before we drift too far into the darker parts of the sea, I'd like to know something simple. Where are you listening from tonight? And what time is it for you right now? Somehow it feels fitting to ask, because as we wander into these quiet regions of the ocean, each of us is meeting this moment from a different place, different light, different hour, and yet we're all stepping into the same stillness together. If you're here to unwind, relax, or let your thoughts settle after a long day, I'm glad you joined this small corner of calm. And if you find this space soothing or helpful, I'd truly appreciate a like or a subscription. It helps bring more listeners to this quieter side of science, where we take topics slowly enough that you can rest while learning about something new. Dead zones might sound dramatic, but they don't begin dramatically. They appear gradually, forming when warm layers of water settle on top of cooler layers and stop the mixing that usually brings oxygen down from the surface. They grow when nutrients from farmland and cities wash into rivers and eventually reach the sea, feeding algae that bloom fast and die faster, leaving behind decay that uses up what little oxygen the deeper water had left. They expand with heat because warm water holds less oxygen to begin with, and currents become lazier, less eager to churn things together. It's a combination of small changes, each one stacking onto the next until the water becomes inhospitable for most sea life. Not empty, not lifeless, but limited in a way that most creatures can't tolerate for long. We won't be speeding through any of this. The ocean doesn't, after all. It takes its time, and life inside these low oxygen areas takes even longer, stretching every moment of energy as far as it will go. That's why easing into this topic feels right. Instead of diving straight into the biology or the chemistry, we're going to wander toward them step by step, the same way the ocean drifts from healthy, oxygen-rich waters into these quieter, strained zones. Imagine the water column as a layered world. At the surface, sunlight passes through and fuels the bustling communities of phytoplankton, the drifting plants of the ocean that feed almost everything else at one level or another. Below them swim countless fish, some in schools, some wandering on their own, all relying on oxygen carried by the currents. Deeper still, light fades, temperatures drop, creatures adjust. But even far down, oxygen usually persists. It may be less plentiful, but it's still there. Enough for slow-moving animals, enough for patient hunters, enough for the countless small life forms that build the scaffolding of the ocean's ecosystems. But as we descend toward a dead zone, something shifts. The water stagnates. The animals thin out. The energy in the environment weakens. You might picture dead zones as quiet rooms at the bottom of the sea, but they aren't always deep. Some stretch across continental shelves. Some hug coastlines where river runoff feeds enormous blooms of algae. Others form in fjords or semi-enclosed seas where water circulation is naturally slow. And some drift through open water, low oxygen pockets floating like underwater deserts. They're different from one another, but they share the same defining drawback. The oxygen drops below what most creatures need to function, and the usual rhythm of marine life fades into something sparse, but sparse doesn't mean empty. And this is where the story becomes interesting, because when the ocean pulls back its oxygen, only the organisms that have learned to work around that challenge remain. Some reduce their energy use until they nearly stop moving. Some change the way they breathe altogether. Some rely on relationships with microorganisms that can tolerate environments most animals would flee. Some drift at the fringes, using the edges of these zones as either feeding grounds or refuge. Nothing here can afford to waste much energy. Everything that survives does so with intention, whether it's a microbe, a worm, a jelly, or an obscure type of fish that has learned to tolerate what most of the sea cannot. When we talk about what survives in places like this, we tend to imagine something eerie or otherworldly, but these organisms aren't strange as much as they are practical. They do exactly what they need to do to get by. In the open ocean, where resources shift constantly and conditions can change in minutes, that practicality is what defines survival. These creatures are specialised, adapted, uncompromising in the way they manage oxygen scarcity, and their existence tells us something important about the resilience of life. Not the dramatic version of resilience we sometimes imagine, but the slow, steady kind that persists quietly, almost unnoticed. It's easy to overlook these organisms because they aren't always the ones we picture when we think of marine life. We tend to imagine dolphins, sharks, coral reefs, bright fish moving in huge schools, but the ocean's stability depends on far more than its vibrant surface dwellers. It depends on the invisible networks happening out of sight, microbes that cycle nutrients even when oxygen disappears, worms that burrow into sediments and push tiny amounts of water through the layers beneath them, jelly-like drifters that move slowly enough to survive in places with nearly no oxygen at all, silent workers that keep the system functioning even when conditions become difficult. Before we move deeper into the details of these organisms and the strategies they use, it helps to picture the scale of the problem. Dead zones have been increasing over the past several decades, expanding in number and size. Climate patterns, warming waters, and nutrient runoff all play roles. And as these zones grow, the creatures that depend on oxygen-rich habitats find fewer places to go. Some migrate to narrower bands near the surface where the oxygen persists. Others try to outrun the conditions, moving along coastlines or deeper into open water, hoping to find new routes where currents still bring in fresh oxygen. But not everything can move that easily. And in the places where mobility is limited, the balance shifts toward the species that don't need oxygen the way most of their neighbours do. What's fascinating is how differently these survivors approach the same challenge. Some reduce their metabolism to a crawl, stretching every bit of energy as far as possible. Some have biochemical tricks that allow them to switch to alternative forms of respiration. Some store oxygen in their tissues when they can find it. Others avoid oxygen altogether, living entirely off chemical processes that don't require it. Some microbes even thrive in these places, not just surviving but using the harshness to their advantage. They perform chemical reactions that shape entire nutrient cycles, reactions that would be impossible in oxygen-rich waters. They become, in a sense, the rulers of these zones. While larger animals may flee, microorganisms take over. But we aren't rushing into those details yet. We'll get to all of them slowly, piece by piece, because that's how understanding builds. These organisms didn't adapt overnight, and we won't rush through their story either. The ocean's steady pace encourages us to match it, to drift carefully from one idea to the next, just as currents drift from one layer of water to another. Nothing here needs to feel abrupt or hurried. It's just a steady walk through a topic that rewards patience. If you've settled in comfortably, you can imagine the sound of distant water, not crashing or rushing, just the faint, constant movement of something enormous shifting beneath the surface. That same movement continues even in the oxygen-poor places we're exploring tonight. It's never truly silent in the ocean. There's always some form of life, some form of rhythm, even if it's slower, quieter, and more fragile than what we're used to thinking about. These dead zones aren't frozen in time. They change with the seasons. They expand and shrink. They shift with the climate. They react to storms, winds, river flows, and the organisms that live in them respond just as steadily, adjusting their behaviour based on tiny cues that most of us would never notice. As we continue, we'll begin examining the first major shift in the water column, the point where oxygen begins to run out and the types of life start to change. But for now, we're still drifting at the edge, noticing how the ocean builds the conditions that create these quiet regions. It's a slow imbalance at first. A little more warmth at the surface, a little less mixing, a few more nutrients washing in from upstream, a bloom of algae that explodes into colour and then sinks into darkness. A stretch of water that grows still, like a room where no one has opened a window in far too long. From there, the oxygen fades. Twice as fast in some places, gradually in others, and once the drop begins, the rest of the change follows. Somewhere beneath the surface, the boundary forms, not a sharp line, but a soft gradient where the rules of survival begin to shift. And that is where our story naturally leads next, continuing downward into the part of the ocean where breath becomes scarce, and the creatures that remain must live with far less than most of their neighbours could ever tolerate. When the water begins to lose its oxygen, the change doesn't unfold with any dramatic signal. There's no sudden shift that would alert a passing animal, no thunderous moment that warns the ecosystem of what's coming. It happens slowly, quietly, and so gradually that many creatures drifting through the upper layers have no sense of the shift until they begin to feel it in their bodies. For us, on land, the idea of running out of breathable air is frightening and immediate. But for the ocean, the shortage builds in a way that feels more like a long sigh than an emergency. To understand what happens in these early stages, it helps to picture the water column not as an empty vertical space but as a layered environment, each part shaped by sunlight, temperature, movement, and the chemistry within it. Near the surface, the ocean is constantly interacting with the air. The wind pulls oxygen down into the upper waters as if stirring ingredients in a bowl. Waves push that oxygen deeper, helping mix the surface layer with what lies beneath it. The sunlight warms the upper water, creating a layer that is slightly lighter and more buoyant than the water below. Usually the cooler, heavier water sinks while the warmer water stays above it, and gentle mixing still happens between them. That constant motion allows oxygen to move downward, often slowly but consistently enough that life throughout the water column gets what it needs. But when conditions begin to shift, that natural mixing becomes disrupted. One of the first things that can disturb this balance is temperature. As surface waters warm more than usual, the top layer becomes even lighter, even more resistant to mixing with the cooler layers beneath. It forms a stable lid that prevents the usual churning of oxygen downward. Even small changes in temperature can amplify this effect, making the warm layer behave like a sealed cover on the ocean surface. Below that cover, the water becomes increasingly isolated. Any oxygen already present slowly gets used up by marine organisms and by chemical reactions involving decaying organic material. Without fresh oxygen coming in from above, the lower layer becomes trapped in a cycle of steady decline. Another contributor to the early stages of oxygen loss is nutrient runoff. When excess nutrients from farming, sewage, or industrial sources enter rivers and eventually reach the sea, they trigger large blooms of phytoplankton. These microscopic plants grow rapidly in the presence of high nutrient levels, sometimes forming dense layers that stretch across many kilometres. At first, this might look like a good thing. After all, phytoplankton form the base of the marine food web. But the speed at which these blooms grow is far beyond what the surrounding ecosystem can handle. When the bloom collapses, millions of dead cells sink to deeper layers of water, where bacteria begin digesting them, and that digestion requires oxygen. The more material there is to break down, the more oxygen the bacteria consume. This creates one of the defining pressures on deeper water. As bacteria work tirelessly to decompose the sinking organic matter, they remove oxygen from the water at a rate much higher than normal. With the surface sealed by warm, buoyant water, and the deeper layers busy processing the remains of the bloom, the balance tips further toward hypoxia, the state of having too little oxygen to support most marine life. The organisms that depend on oxygen for normal activity begin to disperse. Many of them move upward into the thin layer where the wind still manages to supply the water with a steady amount of oxygen. Others migrate horizontally, following currents or coastline features that lead them toward waters with more mixing. But not all marine creatures move quickly or easily. Some are tied to their habitats, some are slow swimmers, and others simply don't sense the shift until the oxygen level falls below what they can tolerate. This point in the process is not yet a dead zone, it's more like the early stage of a long decline. The water still contains oxygen, just not enough for comfort. Fish passing through may begin to show signs of stress. Their gills work harder, their movements become less precise. Some species that rely on high levels of oxygen to fuel their energetic lifestyles are the first to retreat. Predators that hunt by bursts of speed often avoid low-oxygen waters entirely because their bodies cannot function effectively without more oxygen than these waters can provide. Many smaller organisms begin to drift away as well, either actively swimming or relying on currents to carry them to better conditions. As oxygen declines further, the water begins to change in ways that may not be visible, but are deeply significant. Chemical processes that normally occur in oxygen-rich waters slow down or cease altogether. Other processes, which only take place when oxygen levels are extremely low, begin to dominate. In the sediments on the ocean floor, microorganisms begin to switch to different metabolic pathways that do not require oxygen. These microbes use substances like nitrate or sulfate to extract energy instead. This switch introduces new chemicals into the water, some of which can accumulate and create additional challenges for larger marine organisms. Conditions become progressively more hostile to anything that relies on oxygen. In these moments, the first true shift in the ecosystem becomes clear. Animals that typically inhabit the bottom or midwater layers begin to abandon those zones. Crabs and shrimp crawl upward or outward, seeking fresher oxygen. Bottom-dwelling fish rise into shallower water, sometimes crowding into narrow bands near the surface. In severe years, fishermen report seeing large gatherings of fish swimming unusually close to the air-water boundary, appearing almost desperate for more oxygen. Other animals simply leave if they can, moving along the seafloor or through open water until they encounter conditions that better support their needs. Their absence begins to create gaps in the food web, leaving predators with fewer targets and herbivores with fewer grazers to keep algae in check. The decline in oxygen also affects larval stages of many marine creatures. Eggs, larvae and juveniles are often less capable of handling stressful conditions. Even if adults can tolerate low oxygen long enough to escape, the early stages of their species may not survive. This leads to delayed consequences that can affect entire populations years after the initial oxygen loss occurred. Recruitment, the process of new individuals joining the population, becomes unpredictable. Some species may experience a rapid drop in numbers not because the adults died, but because the next generation never had a chance. Despite this steady movement out of affected areas, not everything flees. Some organisms begin to adjust their behaviour, slowing their movements to conserve oxygen. Certain species of jellyfish tolerate low oxygen water far better than fish. They continue drifting, feeding and reproducing, even as the water becomes increasingly inhospitable for other creatures. Their ability to use so little oxygen allows them to thrive where others cannot. This can sometimes lead to temporary increases in jellyfish populations, as they encounter fewer competitors and predators in these dim, strained waters. Microorganisms respond even more quickly to the change. As oxygen levels drop, microbial communities reorganize. Some species disappear from the water column entirely. Others expand, multiplying rapidly because the conditions finally match their specialized metabolisms. These microbes are the first wave of survivors in hypoxic conditions. They take advantage of the chemical environment, using alternative forms of respiration and consuming compounds that would normally remain stable in oxygen-rich waters. Their activity reshapes the chemistry of the entire zone long before larger animals adjust to the transition. This process continues as long as the oxygen remains low and the conditions stay stable. But stability in the ocean is always temporary. Currents may shift, bringing in a small wave of fresher, oxygenated water. A storm might churn the layers and introduce oxygen downward. A change in wind patterns could cool the surface and allow mixing with the deeper layers. These events can temporarily ease the decline, slowing the onset of severe hypoxia. However, if the underlying causes remain, warm surface water, high nutrient input, slow circulation, the decline resumes as soon as the temporary mixing ends. What makes this phase particularly important is that the threshold between low oxygen and dangerously low oxygen is not as large as one might imagine. Once the water reaches a certain point, the decline accelerates. Every bit of organic material that sinks requires bacteria to metabolize it, which consumes even more oxygen. In warm water, bacteria become more active, speeding up the rate at which oxygen disappears. This creates a feedback loop. Warmer water leads to less mixing, which leads to more blooms, which send more organic matter downward, which leads to more bacterial activity, which removes even more oxygen. The deeper layers become deprived not just by chance, but by the mechanics of the ecosystem reacting to an overload of nutrients and a lack of circulation. For the marine life still present in these declining waters, the situation becomes more complicated. Some animals begin using behavior known as habitat compression. Rather than spreading out across a large vertical range, they gather into the narrow band of water that still contains enough oxygen to sustain them. This crowding changes feeding behaviors, predator-prey interactions, and competition. In healthy waters, many species rely on vertical movement to manage their energy use, find food, or avoid predators. When the usable layer shrinks, many of these strategies become impossible. The system becomes packed into a thin, oxygenated strip at the top of the water column. Every creature that cannot dive below or move outward has no choice but to remain in this narrow space, which increases stress on both the organisms and the food web. This stage is where the water begins to feel noticeably different to biologists measuring it. Instruments reveal the profile of the oxygen decline, showing a sharp drop in dissolved oxygen just below the surface layer. In some cases, the drop may seem almost abrupt, even though the process itself has taken weeks or months to develop. The data reveals a zone where oxygen levels fall below 2 mg per litre, the standard threshold for hypoxia. Below that threshold, many species experience difficulty maintaining normal physiological function. Some are unable to regulate their internal chemistry. Others become sluggish, lose coordination, or fail to reproduce. While a small number of species can tolerate this level of oxygen, the majority begin avoiding or abandoning the area entirely. As oxygen continues to decline beyond this point, the water moves toward a condition known as anoxia, where oxygen is nearly absent. But even before it reaches that extreme, the changes in the biological community are unmistakable. What was once a diverse ecosystem with a wide range of species becomes dominated by a handful of organisms that can tolerate or even exploit the low oxygen environment. Microbes multiply. Certain worms persist in the sediments. Some gelatinous zooplankton remain active. But fish, crustaceans, and many invertebrates vanish from the area, leaving behind a quieter, slower world that bears little resemblance to the vibrant waters above. At this stage, the water has begun its transition into what many would call a dead zone, though the term can be misleading. The water is not truly dead, but it has lost most of its familiar inhabitants. It has become a place where only the most adaptable organisms remain. These changes mark the end of the early decline and the beginning of something more complex. The slow disappearance of oxygen sets the stage for a different kind of ecosystem, one that functions on different rules and relies on different processes. The deeper we go into this environment, the clearer it becomes that oxygen is only one part of a much larger interaction between physical forces, biological reactions, and chemical shifts. The water is evolving into a place where survival takes a different form, where the strategies of the organisms that remain no longer resemble those in healthier waters. The story continues as the oxygen falls further and the inhabitants adjust in ways that reveal their remarkable ability to persist even when the ocean provides almost nothing to sustain them. When the ocean slips into a state where oxygen is scarce, the change is not only chemical, but atmospheric in its own way. The water begins to feel muted, as if the energy that usually moves through it has thinned out. It's not a silence you can hear, because of course sound still travels, currents still move, and the biological activities that continue still create their soft disturbances. But on a biological level, something has quieted. Many of the animals that would normally fill the space are no longer present. Their movements, their feeding, their constant low-level activity are gone. What remains feels steadier, slower, and unusually still for something as dynamic as the ocean. This quietness has a practical cause. Creatures that rely on steady supplies of oxygen behave differently when it starts to disappear. They reduce their movement because motion costs energy, and energy requires oxygen to process reliably. When that oxygen is not available, the animal has no choice but to lower its output. Even species that can tolerate lower oxygen levels shift their behaviour. A fish that normally makes sudden, powerful escapes from predators becomes hesitant. A crab that would eagerly explore its surroundings begins to withdraw, saving energy for the essential functions it cannot avoid. This collective reduction in activity creates an environment where almost every organism appears to be holding back, waiting for conditions to improve or for the water to bring something more supportive. The water itself doesn't slow down in a literal sense, but the interactions within it do. Fewer animals are chasing each other. Fewer are grazing the algae or moving through the sediments. But it is not only the absence of familiar marine life that creates this calm. The microorganisms that thrive in these places behave differently too. They may be active, but their activity is microscopic, hard to visualize, and lacking the visible cues that larger animals create. The calm isn't a complete absence of life. It's an absence of the familiar signs of motion that we associate with ocean ecosystems. When scientists observe hypoxic waters, they describe the water column as having an altered structure. It is stratified more sharply than normal. The boundaries between layers are more distinct. Warm water remains above, cooler water sits below, and the mixing between them is minimal. This separation gives the water a layered quality that reinforces the impression of stillness. The layers don't blend easily. They stay in place unless forced by storms or strong winds. In these stratified zones, the calm is not a peaceful state, but a consequence of physical separation within the water column. The ocean becomes partitioned into regions that behave independently rather than working together as a single circulating system. This partitioning also affects how light moves through the water. In oxygen-rich areas where plankton flourish in balanced numbers, the interaction between light and water is dynamic. As phytoplankton grow, die, and move, light is absorbed and scattered. In newly hypoxic areas, the abundance of decaying organic matter and the changes in microbial communities can alter how light penetrates the surface. Sometimes the water becomes murkier, with a brown or green tint from the remnants of algal blooms. In other places, the water beneath the surface takes on a darker, more stagnant appearance, as if the light is struggling to reach the depths where oxygen has vanished. biologists who study hypoxia often speak of the water having a flat character during these conditions. That word may sound imprecise, but it reflects a real observation. When fish schools are absent, when bottom dwellers have fled or died, and when the variety of motion within the water column diminishes, the ocean feels visually simplified. There are fewer layers of movement, fewer reactions, fewer small shifts that usually create the sense of a living system. In this quietness, the organisms that remain stand out more sharply, even though they themselves may be moving very little. Some of the creatures that linger in these areas give the calm a slightly eerie quality. Certain jellyfish, for example, drift through low oxygen waters with an ease that other animals lack. Their slow pulsing movement remains nearly unchanged even when oxygen levels drop. Because they do not rely heavily on oxygen for their metabolic processes, they continue to drift, feed, and reproduce while fish retreat from the area. Their presence adds to the perception of stillness. They move at a deliberate, steady pace, their bodies rising and falling with the faintest current. No sudden motions come from them, no quick reactions. They appear unbothered by the chemical shift around them, creating a contrast with the animals that cannot stay. Worms living within the sediments also contribute to the calm. In oxygen-rich waters, many of these animals constantly process sediment, poking in and out of the substrate and stirring the material beneath them. In hypoxic conditions, only the species that can tolerate low oxygen remain, and even they reduce their activity. They digest material more slowly. They move less frequently. They leave fewer signs of disturbance along the sea floor. The sediment becomes smoother and less textured, lacking the typical patterns that active benthic life creates. This smoothness reinforces the impression that the environment has grown still. Microbial mats often form on the seafloor under prolonged hypoxia. These mats, though alive, create a unified surface appearance that hides the small-scale movements below. They're composed of dense colonies of bacteria that thrive on chemical reactions involving sulfur, nitrogen, or other compounds present in low-oxygen environments. These mats sometimes appear as pale films, yellowish layers, or cloudy white patches across the bottom. Their presence is another contributor to the quiet, muted appearance of hypoxic seas. They cover the ground where sea stars, clams, and mobile crustaceans would normally leave traces of their activities. Instead, everything appears coated in a thin layer that gives the impression of inactivity. Another aspect of the calm is the change in predator-prey dynamics. In healthy waters, predators chase prey. Prey attempt to evade them. These interactions generate bursts of movement and energy. In hypoxic regions, these patterns collapse. Fish capable of leaving do so, reducing the number of hunting opportunities. Many predators refuse to enter the area at all, because pursuing prey requires bursts of activity they cannot maintain without oxygen. Meanwhile, the prey that remain are often low-oxygen-tolerant species that do not attract the same kinds of predators. This simplified interaction reduces one of the major sources of motion in the water column. Without the constant cycle of pursuit and escape, the region grows quieter in every sense of the word. The calm is further emphasized by the absence of sound generated by certain marine animals. Many fish produce noises, whether through grinding their teeth, vibrating specialized organs, or moving water through their gill covers. Crustaceans like snapping shrimp create sharp, clicking sounds. When these species leave hypoxic regions, those sounds disappear. Hydrophone recordings show noticeable differences between oxygen-rich areas and hypoxic ones. The latter contain fewer sharp noises, fewer repetitive clicks, and fewer hums created by busy animal communities. What remains are the low, continuous tones produced by distant currents or geological features. The biological contributions to ocean soundscape diminish, adding to the overall sense of emptiness. Chemical changes in hypoxic water can add specific odours or tastes noticeable to marine animals, though not to humans at the surface. Some species detect these chemical signals long before oxygen becomes dangerously low. They interpret them as warnings, prompting them to leave. The organisms that stay are either unable to detect the shift, unable to leave, or adapted to conditions in which these chemical changes mean something different to them. The calmness that follows is partly due to the departure of animals sensitive to these cues. Because their sensory warnings push them away, the waters become populated only by those who ignore or tolerate the shift, and their behaviour does not produce much visible activity. This calmness can deceive observers who might assume the area is empty, but emptiness is not the true state of hypoxic seas. The remaining organisms include bacteria performing intricate chemical cycles, certain types of plankton drifting slowly, and a few low-oxygen-tolerant animals maintaining minimal movement. These activities continue out of sight for the most part. While quiet, they are crucial to the way the region develops. The chemical transformations happening in this phase set the stage for the deeper changes that occur when oxygen levels drop even further, reshaping the region into a specialized environment that supports only a handful of species. The calm of hypoxic seas also creates a transitional zone between habitable and uninhabitable water. Fish that approach the boundary often hesitate, sensing the low oxygen with receptors in their gills. They may swim along the edge, repeatedly testing the water before turning back. This behaviour is seen in many species and contributes to a concentrated band of activity at the upper boundary of the hypoxic layer. Above that boundary, animals remain active. Below it, their numbers drop sharply. The boundary acts like a dividing line that separates movement from stillness. Observers sometimes describe this as if the ocean had placed an invisible ceiling beneath which the usual inhabitants refuse to descend. The result is an unusual contrast between the turbulent life above and the subdued world below. The longer hypoxic conditions persist, the more pronounced the calm becomes. Sediments accumulate organic material without the usual processing from active bottom dwellers. Microbial films expand. Jellyfish drift through open waters that would normally be full of fish. Small crustaceans that can tolerate low oxygen move slowly along the bottom, and their reduced activity contributes further to the sense of stillness. Because these organisms often have slower metabolisms, their movements follow a consistent, steady pattern rather than the unpredictable bursts typical of many marine species. This gives the entire region a measured, unvarying quality that stands out from the usual variability of ocean life. Even the currents interact differently with this environment. In healthy waters, currents can trigger a chain reaction of activity. They can sweep plankton into new areas, prompting feeding events. They can push sediment along the bottom, stirring up organisms hiding there. In hypoxic waters, these reactions are muted. Currents continue to move, but there are fewer creatures responding immediately to these shifts. The water's motion does not translate into biological motion in the same way. This disconnect between physical forces and biological response makes the region feel detached from the usual ocean rhythms. Hypoxic seas can persist for weeks, months, or even years depending on the region. During their longest phases, they begin to take on predictable traits beyond the calm itself. The remaining organisms settle into patterns that become routine for them. Microbes running anaerobic processes become the dominant life forms. Jellies use the open space to feed with less competition. Certain small worms and crustaceans adjust to the conditions and continue their slow activity. Over time, these patterns reinforce the calm and reduce the likelihood of rapid ecological rebound. If oxygen returns suddenly, many of these organisms will adjust again, but while hypoxia remains, they define the environment. This calm is not a sign of stability. It is the visible surface of a system under stress. The lack of movement, the minimal interactions, and the reduced diversity are signals that the region is unable to support the level of life it once held. The calmness becomes a kind of quiet warning. Once the water reaches a point where it no longer sustains active communities, reversing that condition requires either large-scale physical mixing or the reduction of nutrient input that triggered the hypoxia in the first place. Without intervention, the calm persists and may deepen toward a more severe state, in which the water approaches or reaches complete anoxia. As this phase continues, it becomes clearer why certain organisms survive when others cannot. Their metabolic strategies, tolerance levels, and ecological roles determine their chances of remaining within these waters. The calm of hypoxic seas provides the setting in which these differences appear most dramatically. It is within this quiet environment that the next stage of adaptation begins to emerge, revealing how life adjusts when oxygen is no longer guaranteed, and survival requires entirely new strategies shaped by scarcity and endurance. Some of the most revealing moments in a hypoxic region come from watching the animals that refuse to leave even after the oxygen has dropped to levels most creatures would struggle with. These are not always dramatic or imposing species. They're often small, sometimes slow, and occasionally dismissed in healthier ecosystems because they tend to hide, burrow, or drift without drawing much attention. But in the quiet of low oxygen waters, their presence becomes a kind of signature. They remain where others have fled, holding on to whatever small opportunities the ecosystem still offers. Their persistence is not rooted in strength or size, but in an array of physical and behavioural adjustments that allow them to function in environments many animals would avoid altogether. One of the first groups to consider are fish that have evolved the capacity to tolerate short periods of low oxygen, often by modifying their behaviour before modifying their physiology. Some of these species approach hypoxic water cautiously, sensing the shortage and adjusting their movements in ways that reduce oxygen demand. For example, certain species of croaker and drumfish continue to occupy hypoxic waters longer than other fish because they shift from active swimming to slow gliding. When their bodies no longer require rapid bursts of energy, they rely less heavily on the fast-twitch muscle fibres that need the most oxygen. This subtle shift in behaviour allows them to maintain a presence in areas where other species begin to struggle. These fish also tend to reduce activities that would normally expend energy, such as aggressive displays, territorial defence, or courtship movements. Even though they stay in the region, they do so in a way that minimises the cost of remaining there. Another group known for enduring low oxygen conditions are certain flatfish species. They're often associated with seafloors in coastal areas, and some have developed strong tolerance for oxygen levels that would challenge most other fish. Part of their success is tied to how they live. Instead of swimming in the water column, they rest along the sediment where slight differences in oxygen levels might give them a small advantage. Some microcurrents created by subtle topographic features on the seafloor can bring faint traces of oxygen to their gill area. By remaining still and positioning themselves carefully, these fish can survive longer in hypoxic zones than those that rely on constant motion. Their gill structures are also adapted to maximise oxygen extraction, allowing them to absorb more from the water even when the supply is extremely low. A different strategy appears in some species of catfish and eels living in estuarine environments. These animals utilise the ability to gulp air at the surface when needed. Even in marine or brackish waters, some of them rise periodically to take in oxygen directly from the atmosphere. Their bodies contain vascular structures capable of absorbing oxygen through specialised tissues, supplementing what they draw from the water. This dual strategy allows them to inhabit waters that many other species would consider unlivable. These animals are not dependent on this ability in normal situations, but when hypoxia develops, the option to access air gives them a significant advantage. Beyond fish, crustaceans provide some of the most interesting examples of creatures refusing to give up on oxygen-depleted environments. Many shrimp species cannot tolerate hypoxia and flee early, but a few are equipped with strategies that let them remain longer than expected. Some species reduce their metabolic rate dramatically, almost entering a slowed physiological state to conserve the oxygen in their bodies. Their gills are efficient at extracting oxygen, and in some cases they can survive in waters where oxygen levels fall below the threshold that would kill less adaptable crustaceans. Their continued presence maintains part of the lower food web, though their reduced movement changes how they interact with their surroundings. Crabs show an even wider range of responses to low oxygen. Blue crabs, for example, can tolerate a surprising degree of hypoxia for short periods by retreating into shallow waters where oxygen from the atmosphere diffuses into the surface layer. They adjust their behaviour by moving less and reducing their feeding. At extremely low oxygen levels, they switch to anaerobic metabolic pathways that allow them to sustain basic functions for limited times. Although anaerobic metabolism is inefficient and cannot support them indefinitely, it gives them enough time to survive short hypoxic events until mixing or circulation returns. Other crab species take advantage of burrows they dig into the sediment. While the sediment itself can become hypoxic or even anoxic, some burrows allow small threads of oxygenated water to enter, creating pockets where crabs can endure longer than they would in the open water. Mollusks also appear frequently among the species that resist leaving hypoxic areas. Some bivalves, such as clams and oysters, are sessile organisms that cannot simply swim away from low oxygen. Their persistence is not optional, but necessary, and their survival depends on physiological flexibility. Certain clam species reduce their metabolic rate dramatically, lowering their oxygen requirements by slowing their feeding and filtration processes. Some can even close their shells for extended periods, trapping a small amount of oxygen-rich water inside. Although this strategy cannot support them indefinitely, it allows them to endure episodes of low oxygen until conditions improve. Mussels have similar capabilities, though their tolerance thresholds vary widely depending on species and environment. A few species of snails living near the seafloor also show resilience in low oxygen environments. Their metabolic demands are generally lower than those of more active animals, and some have the ability to store oxygen in hemocyanin-rich blood that carries higher oxygen binding capacity. While they still experience stress in hypoxic waters, their ability to slow movement and their naturally low respiratory demands give them an advantage. Because they often feed on detritus or slow-growing algal films, they can continue feeding even when other animals have left the area, maintaining at least part of the benthic nutrient cycle. An important group of organisms that endure hypoxia are polychaete worms. These segmented worms occupy sediments in a wide range of marine habitats. Many species have gills or parapodia rich in blood vessels that allow them to absorb oxygen efficiently, even in low concentrations. Some species adjust by burrowing closer to the surface of sediments where oxygen penetration is slightly higher. Others shift to anaerobic metabolic pathways temporarily. Polychaetes often dominate hypoxic sediments because their adaptations allow them to persist where less flexible species cannot. Their continued activity helps sustain sediment turnover, even at reduced rates, preventing the seafloor from becoming entirely inactive. The group that shows the most impressive array of adaptations, however, are microorganisms. Bacteria and archaea capable of surviving without oxygen are common in many marine environments, but their role becomes dominant in hypoxic and anoxic zones. Some bacteria switch from oxygen-based metabolism to nitrate reduction, sulfate reduction, or even the use of metals like iron or manganese as electron acceptors. These processes create byproducts such as hydrogen sulfide, which can accumulate in the water and sediment. Despite this, the bacteria continue to thrive because their metabolic pathways are designed to operate efficiently in the absence of oxygen. Their presence shapes the chemical landscape of hypoxic areas and often determines which larger organisms can tolerate the surrounding conditions. Other microbial groups specialize in detoxifying compounds produced in low oxygen environments. For example, sulfur-oxidizing bacteria use hydrogen sulfide generated by anaerobic microbes as a source of energy, converting it into less harmful substances. These bacteria form dense mats in some hypoxic and anoxic zones, creating visible layers along the seafloor. While these mats might appear lifeless to the casual observer, they are actually signs of thriving microbial communities. Their activity not only keeps certain harmful compounds in check, but also provides a form of primary production in environments where traditional photosynthesis is not possible. In some cases, microbes and larger organisms form partnerships to survive low oxygen conditions. Certain bivalves host symbiotic bacteria inside their tissues. These bacteria process chemicals from the surrounding water or sediment, providing nutrients to the host animal in exchange for shelter. These partnerships allow the host organisms to survive in places where they would otherwise be unable to feed effectively. The presence of symbionts gives them access to chemical energy sources unavailable to animals, relying on oxygen-driven metabolic pathways. Even within the water column, certain zooplankton species show unexpected tolerance for hypoxic environments. Some copepods, for instance, have been observed moving within low oxygen layers for brief periods. They do not remain there permanently, but their ability to enter and leave these waters allows them to exploit food sources that most other zooplankton avoid. Their movements through the boundary layers between oxygen-rich and oxygen-poor water contribute to small-scale mixing, influencing nutrient distribution. Some species accumulate oxygen in their bodies while they are in more oxygenated waters, storing enough to allow them to remain below the threshold for short intervals. This strategy gives them access to regions where predation pressure is lower because so many predators cannot enter. Jellyfish remain one of the most recognizable creatures that persist in hypoxic seas. Their simple body structure, low metabolic requirements, and reliance on passive feeding allow them to thrive in conditions that make life difficult for many other species. Jellies do not require much oxygen to maintain their buoyancy or movement. Their tissues absorb what they need directly from the water, and because their oxygen demands are modest, they can continue moving slowly through the water column long after fish have either retreated or died. Their ability to exploit these conditions can lead to temporary increases in jellyfish numbers, since competition for food is reduced. This phenomenon has been observed in several regions affected by hypoxia, including parts of the Gulf of Mexico and the Baltic Sea. Some species of squid show remarkable short-term tolerance to hypoxic waters by regulating their buoyancy and reducing activity. These are typically smaller species, not the large active squid associated with deep dives and high-speed hunting. They slow their jet propulsion, hover more often, and depend on ambush tactics rather than active pursuit. Their tolerance is limited, but it allows them to persist in mildly hypoxic regions where larger predators withdraw. When oxygen drops too low, they leave the area quickly, but their initial resistance places them among the group that holds on longer than many fish. Several species of small fish known as killerfish demonstrate a more advanced form of hypoxia tolerance. They can switch to anaerobic metabolism for longer periods than many marine species. Additionally, their bodies can alter the expression of certain proteins involved in oxygen transport, giving them increased efficiency when oxygen becomes scarce. Some killerfish populations live in estuaries where hypoxia is a regular seasonal event, allowing them to develop both physiological and genetic adaptations over time. These fish often serve as model organisms for understanding how vertebrates respond to low oxygen environments. Even coral reefs, though often highly sensitive to water quality changes, contain a few coral species that tolerate short-term hypoxia. These corals slow their respiration, rely more heavily on stored energy, and reduce the activities of their symbiotic algae. Their tolerance is limited, but in certain lagoons and reef flats, hypoxic episodes occur regularly, and these corals have adapted to endure temporary reductions in oxygen. Their survival does not mean the reef remains healthy, but their persistence shows that some reef organisms have more flexibility than expected. The creatures that remain in hypoxic waters are not defined by a single trait. Their success comes from a combination of physiological capacity, behavioral adjustments, and ecological positioning. Some rely on storing oxygen, some depend on reduced metabolism, some access air when needed. Some form symbiotic relationships that remove them from the constraints of oxygen-dependent feeding. These strategies reveal how varied the responses to hypoxia can be. They also show that low oxygen environments, while stressful, do not eliminate life entirely. Instead, they filter the ecosystem, leaving behind only the species equipped to endure scarcity. Understanding these organisms helps explain what happens next in hypoxic seas. As oxygen continues to decline, the balance shifts further towards species with anaerobic metabolic pathways or chemical-based energy strategies. The organisms discussed here represent the final group that can still function with limited oxygen, before the environment transitions into something even more restrictive. Their resilience forms the bridge between hypoxic waters and the deeper, more extreme conditions that follow when oxygen disappears almost completely, shaping the next stage of how life adjusts to the challenges of a changing ocean. In low oxygen regions where most animals flee or falter, the organisms that remain often share one remarkable trait. They have mastered a way of living that uses as little energy as possible. Instead of relying on speed, constant feeding, or rapid responses to changes around them, these creatures survive by reducing every unnecessary cost. In an environment where oxygen defines the limits of activity and food may become scarce, their entire lives revolve around conserving resources. To understand how they function, it helps to picture an ecosystem where the rules have shifted. Movement is expensive, growth is slow, feeding is unpredictable. Everything depends on stretching limited energy across long spans of time. Many animals that stay in hypoxic waters do so not because they are robust in the usual sense, but because they are efficient. Their energy budgets are designed to support slow living. One of the primary adjustments seen in these organisms is a drop in metabolic rate. By slowing the rate at which their cells consume energy, they reduce their need for oxygen. This isn't a temporary response triggered only during severe stress. In many cases, their baseline metabolism is naturally low, giving them an advantage whenever oxygen becomes scarce. Some species reduce their metabolic activity even further when conditions worsen, entering a sustained state where only essential functions continue. Worms living in near-anoxic sediments are good examples of how slow living works in practice. Many polychaete worms found in hypoxic zones have evolved to function with minimal oxygen intake. Their bodies are slim, allowing oxygen to diffuse across a larger relative surface. They move slowly and deliberately in the sediment, limiting their muscle contractions to the smallest motions needed for feeding or repositioning. These worms often rely on stored energy reserves, using them gradually. When food becomes available, they feed efficiently, but they do not depend on a constant supply. Their digestive systems can handle low-quality organic matter that other species may ignore, allowing them to thrive on leftovers within the environment. Some bivalves use an even more strategic approach. When oxygen drops, they close their shells tightly, keeping only the bare minimum of water inside. By sealing themselves, they create a microenvironment that contains slightly more oxygen than the surrounding water. Inside the shell, metabolic activity slows dramatically. These organisms may remain closed for hours or even days, depending on the oxygen levels and the temperature. While sealed, they rely entirely on anaerobic metabolic pathways, breaking down stored compounds in a way that allows them to function temporarily without new oxygen coming in. The trade-off is a build-up of metabolic byproducts, but once conditions improve, they open and allow the fresh water to flush these waste products away. Gastropods in these regions take a similar approach. Many small snails reduce their feeding frequency and movement, conserving energy by relying on stored resources. Snails have slower metabolisms by default, and in hypoxic environments this feature becomes a significant advantage. Their feeding strategy, which often involves scraping films from surfaces or consuming soft organic material from sediments, does not require rapid motion. They can remain nearly still for long periods before resuming gentle movements that use minimal oxygen. Some crustaceans embrace slow living with surprising precision. A few amphipod and isopod species reduce their activity during oxygen decline, exhibiting long periods of near immobility. Their bodies are efficient at absorbing small amounts of oxygen from the water, and they can continue minimal activity even when their environment becomes severely hypoxic. Their feeding habits often centre on detritus, which remains available even after fish and larger animals leave. This gives them a stable if limited food source that supports their low-energy lifestyle. Large animals rarely fit the slow-living strategy because their bodies require more oxygen to support tissue maintenance and movement. Yet there are exceptions. Some species of sea cucumbers living in low-oxygen basins have evolved to reduce their activity to almost imperceptible levels. These animals sift through sediment slowly, taking in small amounts of organic matter. Their bodies can tolerate long periods of low oxygen exposure by moderating their internal chemical processes. Sea cucumbers have a fluid body structure that requires little muscular effort to maintain, making them surprisingly resilient in harsh conditions. Another example comes from certain species of brittle stars. Although many brittle stars leave hypoxic areas, some tolerate low oxygen by reducing their feeding and limiting the use of their arms. Instead of crawling actively over the sea floor, they anchor themselves in a stable location and wait for organic particles to drift near their arms. Their tube feet move slowly to capture bits of food, and their internal processes adjust accordingly. They do not grow quickly or reproduce at high rates, but they survive by minimizing their demands on the environment. Jellyfish, though often associated with drifting rather than active locomotion, are prime examples of organisms that naturally use very little energy. Their bodies consist largely of water and require minimal oxygen to maintain their basic life processes. They pulse slowly through the water, letting currents carry them rather than forcing movement. Their feeding strategy relies on tentacles that passively capture prey. This combination of traits allows them to persist in environments where most fish cannot remain. In low-oxygen seas, jellyfish may become some of the most active organisms simply because others have slowed down even more. Their presence in hypoxic waters is not a sign of thriving conditions, but an indication of which species can tolerate the demands of the region without exceeding their oxygen limits. Microbial life also excels at slow living. Many bacteria in low-oxygen regions have evolved metabolic pathways that operate efficiently under minimal energy availability. Some use sulfate or nitrate instead of oxygen, switching to biochemical processes that yield far less energy per molecule but require no oxygen. Their growth rates slow accordingly. These microbes may divide infrequently, maintain small cell sizes, and use available nutrients cautiously. Even so, they remain the dominant force driving chemical changes within hypoxic environments. Their slow, consistent activity ensures that organic matter continues to decompose, though at a pace far different from oxygen-rich conditions. One of the most striking examples of slow living occurs in microbial mats that develop on the seafloor. These mats are dense communities of microorganisms layered on top of each other. The bacteria at the surface perform one set of reactions, often involving the oxidation of sulfur compounds. The deeper layers house bacteria that rely on compounds produced above them. The entire system runs on slow but steady chemical exchanges. Even though each individual cell has low energy requirements, the collective output of the mat significantly influences the surrounding environment. These mats can persist for long periods, providing stability in areas where most other forms of life struggle. Some species of small, slow-growing sponges are also able to tolerate low oxygen conditions better than expected. Their filtration systems can operate at reduced rates, drawing small amounts of water through their bodies. When oxygen levels drop, they reduce pumping to conserve energy. Sponges have symbiotic relationships with various bacteria, some of which carry out metabolic functions that assist the host. These microbial partners allow the sponge to access nutrients through chemical processes that require less oxygen than typical metabolic pathways. As a result, sponges can persist in areas where many larger animals would not survive. In long-lasting hypoxic zones, different species show staggered patterns of endurance. Some organisms remain active for only short intervals before resting. Others maintain slow but constant activity. These staggered strategies allow multiple species to coexist even in limited space. For instance, a slow-grazing snail may feed intermittently, moving only occasionally. A nearby polychaete worm may remain buried for hours at a time, emerging only when needed to gather food. A crustacean may remain nearly motionless, conserving oxygen, until something small drifts close enough to grasp. Together, these organisms form a network of slow interactions that keep the ecosystem functioning in a highly altered state. Feeding behaviour also shifts dramatically under hypoxia. Many organisms reduce their need for food by decreasing their metabolic demands. Those that do feed often rely on detritus and sediment-associated organic matter, which accumulates due to the lack of larger animals consuming it. Because the rate of decay slows when oxygen is low, detritus may persist longer than usual. This gives slow-moving animals a steady if modest food source. Their ability to process poor-quality organic matter is a defining trait of low-oxygen survivors. These feeding strategies also limit their involvement in active food webs, reducing predator-prey interactions and keeping the system stable at low energy levels. Reproduction in slow-living organisms also reflects their environment. Many species produce fewer offspring during hypoxic conditions, investing more energy into maintaining basic functions. When they do reproduce, they may release eggs or larvae at times when oxygen levels rise temporarily, allowing the next generation a better chance of survival. Some species produce offspring that can disperse quickly to find more favourable conditions. Others create larvae that are more resistant to low oxygen, though these adaptations vary widely across species. Even communication shifts. Many animals rely on chemical signals to find mates, detect predators, or locate food. In hypoxic waters, the diffusion of these chemicals changes. Some signals degrade more slowly due to reduced microbial activity, while others may spread differently because of stratified water layers. Slow-living animals adapt by relying less on fast chemical communication and more on passive cues such as the movement of water or contact with sediment. Their sensory systems do not shut down completely, but they adjust to the altered environment in ways that complement their limited energy budgets. The physiology of slow-living animals contains specific adaptations that support this lifestyle. Some species possess proteins that bind oxygen more strongly, allowing them to extract as much oxygen as possible from the surrounding water. Others increase the number of mitochondria in their cells, improving their ability to generate small amounts of energy under low oxygen conditions. Still others adjust the fluidity of their cell membranes to reduce energy leakage. These cellular details support the broader strategy of surviving on minimal inputs. Behaviourally, many organisms adopt patterns that reduce risk. In oxygen-rich seas, animals may move widely to find food or mates. In hypoxic waters, extensive movement can be fatal. Slow-living creatures often remain in restricted areas, exploring only when necessary. They learn the microhabitats where slight variations in oxygen levels occur, positioning themselves carefully within those narrow windows. This behaviour reflects a finely tuned sense of their surroundings, developed through generations of adaptation. The slow pace of life in a hypoxic environment also affects the flow of nutrients. In oxygen-rich waters, active animals stir sediments, consume material, and recycle nutrients quickly. In low oxygen areas, this process pauses. Nutrient cycling continues, but at a speed dictated by the microorganisms and slow-moving animals that dominate the ecosystem. Their steady actions maintain a baseline of biological processing that prevents the area from becoming completely static. Sediments accumulate material, but they also house the bacteria and worms that break it down. Despite the reduced pace, these processes remain vital for keeping the environment functional. These organisms also serve as the final anchors of life before the transition into anoxic zones. Their adaptations allow them to survive where oxygen exists only in faint traces. They form the last line of oxygen-dependent life, bridging the gap between habitats that support a variety of species and those that support only microorganisms using anaerobic pathways. Their persistence shapes the boundary between these two worlds, influencing how the environment transforms as oxygen levels continue to decline. Understanding how these organisms operate gives insight into the resilience of life under stress. Their strategies are not built on strength or speed, but on restraint, efficiency, and careful use of resources. They represent the culmination of countless adjustments shaped by the pressures of their environment. As conditions change, these creatures do not resist the shift by intensifying their efforts. They adapt by reducing their demands, accepting the limitations imposed by the water, and finding ways to exist within those limits. Their success provides the foundation for understanding the next stage of hypoxic ecosystems. As oxygen becomes even scarcer and the environment moves toward complete anoxia, the organisms that rely on slow living give way to those that no longer depend on oxygen at all. The pace of life changes again, driven by chemical reactions rather than oxygen-based metabolism. The transition from slow living to anaerobic survival marks another shift in how the ecosystem functions, setting the stage for the distinct communities that follow. In the dim layers of water where oxygen falls away and larger creatures retreat, a different world takes shape. It is not marked by fast movement, colourful displays, or the familiar interactions seen in well-oxygenated seas. Instead, the activity shifts to scales too small for the human eye to detect. This is where vast microbial communities take over. Their presence may seem subtle, but their impact shapes almost every chemical process in hypoxic zones. These microorganisms do not rely on oxygen as their main energy source, and in many cases they function better when oxygen is nearly absent. Their strategies are not the adaptations of panic or temporary survival. They are the product of long evolutionary histories that prepared them for environments where oxygen is unreliable. When oxygen begins to diminish, the first group of microbes to expand are those that can shift between oxygen-based and oxygen-free metabolism. They use oxygen when it's available, and when it disappears they switch to alternative pathways. This flexibility allows them to dominate the early stages of hypoxia. Their numbers grow as the competition from other microorganisms declines. These microbes are often the bridge between the last traces of oxygen use and the onset of conditions where only anaerobic processes can occur. They occupy the intermediate layer between the healthier waters above and the deeper, oxygen-stripped waters below, shaping the transition zone with a variety of chemical reactions. As the oxygen drops closer to zero, other groups become more prominent. These microorganisms rely entirely on chemical processes that do not involve oxygen. Some of them use nitrate, a compound naturally present in seawater, as a substitute for oxygen. Nitrate reduction produces nitrogen gas, which bubbles out of the ecosystem. This process removes nitrogen from the water, influencing nutrient cycles in ways that extend far beyond the boundaries of the hypoxic zone. The removal of nitrogen reduces the amount available for algal growth, yet it rarely offsets the large inputs from runoff and decaying organic matter. Still, the activity of these microbes forms a critical part of how hypoxic waters evolve chemically. Another major group includes sulfate-reducing bacteria. These microorganisms use sulfate instead of oxygen or nitrate. Sulfate is abundant in seawater, and these bacteria can thrive in deep sediments or stagnant water where oxygen has vanished completely. Their metabolic activity produces hydrogen sulfide, a compound known for its distinct smell and its toxicity to many forms of life. Hydrogen sulfide accumulates when water circulation is weak, and under certain conditions it builds up to concentrations that could be harmful if released rapidly. The presence of hydrogen sulfide in sediments is often a sign that sulfate-reducing bacteria dominate the deeper layers of hypoxic or anoxic regions. The hydrogen sulfide produced by these microbes becomes the energy source for yet another microbial group. Sulfur-oxidizing bacteria take advantage of this compound, using it to fuel their metabolic processes. They convert hydrogen sulfide into sulfate again, often producing visible mats along the seafloor, or in layers where oxygen and hydrogen sulfide meet. These mats can be pale, yellowish, or white, and their spread indicates that sulfur cycling has become the primary mode of life in the region. The presence of these mats is a clear sign that the microbial empires in hypoxic zones are not only diverse but intricately connected. The activity of one group feeds another, and these relationships drive the stability of the ecosystem in ways that larger animals cannot replicate. In many hypoxic zones, the water column becomes stratified into layers defined not by temperature or salinity alone, but by microbial activity. Near the top of the hypoxic layer, bacteria that can tolerate small amounts of oxygen thrive. Slightly below them, nitrate-reducing bacteria dominate. Deeper still, sulfate-reducing and sulfur-oxidizing communities take over, creating a gradient of chemical processes stacked like invisible tiers. Each layer has a distinct combination of chemical reactions that set it apart from the layers above and below. Because these microbes respond directly to the availability of different compounds, they organize themselves into patterns that reflect the chemical structure of the water. This organization may seem spontaneous, but it is the product of continuous interactions. As one group of microbes processes a compound, they produce byproducts that serve as resources for another group. If nitrate-reducing bacteria consume available nitrate, their waste products may accumulate, influencing how the next group functions. Sulfate-reducing bacteria rely on the presence of organic carbon, which comes from decaying organisms or the remains of algae. When their activity intensifies, they produce hydrogen sulfide, which immediately supports the sulfur oxidizers. The entire system is anchored in a cycle where no single organism stands alone. Each contributes to the larger process of maintaining chemical balance in the absence of oxygen. Some microorganisms in hypoxic zones use even more unusual strategies. Certain bacteria can use iron or manganese in their metabolic processes, oxidizing or reducing these metals as energy sources. These reactions create distinct layers of chemically modified metals within sediments. Over time, iron-rich layers can build up in hypoxic basins, reflecting long periods of low oxygen conditions. The microbes responsible for these reactions play essential roles in shaping sediment deposits, influencing how nutrients and trace metals move through the environment. Although their activity is complex, it represents another example of how adaptable microbial life can be when faced with limited oxygen. Archaea also play significant roles in hypoxic and anoxic environments. These microorganisms are distinct from bacteria and possess unique metabolic capabilities. Some archaea produce methane from organic matter, using a process known as methanogenesis. Methane production occurs in the deepest, most oxygen-depleted parts of the sediment where other energy sources are exhausted. These methane-producing archaea form the endpoint of many chemical reactions in anoxic environments. They operate slowly, often requiring long periods to produce significant amounts of methane. Yet their presence indicates that the microbial community has shifted into a mode where even the most reduced chemical reactions are underway. Methane does not always remain in the sediments. Some bacteria can consume methane, using it as their primary energy source. These methane-oxidizing bacteria occupy layers just above the methanogenic archaea. When methane diffuses upward, it encounters these bacteria, which convert it into carbon dioxide or other compounds. This interaction between methane producers and methane consumers creates another cycle within the system, stabilizing conditions and preventing large amounts of methane from entering the water column. The presence of these paired communities shows how deeply interconnected the microbial processes are in oxygen-depleted waters. Microbial mats represent some of the most visually striking features in hypoxic and anoxic environments. These mats form dense layers of bacteria that accumulate on the seafloor or attach to underwater surfaces. Their structure can vary from thin veneers to thick cohesive sheets, several centimeters deep. They often contain multiple layers of different microorganisms, each specializing in a particular chemical process. The upper layers may contain bacteria that tolerate small amounts of light and oxygen, while the deeper layers host sulfate-reducing bacteria or other anaerobic organisms. These mats can persist for long periods, growing slowly but consistently. They contribute to sediment stability, and serve as microhabitats for small animals that can endure low oxygen conditions. In some regions, microbial mats create a stable foundation for entire microecosystems. Small worms, crustaceans, and other benthic organisms feed on the surface of the mats, grazing on the bacteria or consuming organic material trapped within them. These grazers are often slow-moving, reflecting the broader pattern of low energy living in hypoxic zones. Their interactions with the mats help shape the distribution of microbial communities, although their impact is minor compared to the overwhelming influence of the microbes themselves. The dominance of microbial life in hypoxic areas is not limited to the seafloor. The water column also contains floating aggregates known as marine snow. This material consists of decaying organic matter, dead plankton, fecal pellets, and other particles that slowly sink through the water. As marine snow drifts downward, it carries with it a community of microorganisms that continue breaking down the material. When the particles pass through hypoxic layers, the microbes adjust their metabolic processes to match local conditions. By the time marine snow reaches the seafloor, it has often undergone several stages of microbial processing. These particles are essential sources of carbon and nutrients in low oxygen regions, supporting the microbial communities that dominate there. Some hypoxic zones have persistent water masses that remain isolated from the rest of the ocean for long periods. In these places, microbial communities evolve unique characteristics. Because outside factors such as temperature and salinity remain relatively stable, the chemical structure of the water becomes shaped almost entirely by microbial processes. These isolated systems develop distinctive layers enriched with compounds like ammonia, nitrite, iron sulfide, or methane. Microbes in these areas become highly specialized, often relying on narrow metabolic pathways tailored to the precise chemical environment around them. The stability of these systems allows rare or ancient microbial lineages to persist, providing valuable insights into how life can adapt to extreme conditions. Microbial life in hypoxic zones also influences the global carbon cycle. By processing organic matter that would otherwise accumulate, these microbes help regulate the flow of carbon through the ocean. Some pathways convert organic carbon into inorganic forms. Others produce gases such as methane or nitrous oxide, which can escape into the atmosphere under certain conditions. Although the release of these gases is limited in stable hypoxic zones, disturbances such as storms or upwelling events can bring deeper water to the surface, releasing trapped gases. The microbial activity leading to these releases reflects the long-term impact of hypoxia on global climate processes, even if the immediate effects remain subtle. One of the most surprising aspects of microbial life in hypoxic waters is how quickly these communities respond to environmental changes. When oxygen becomes available, some microbes switch back to oxygen-based metabolism almost immediately. Others withdraw into dormancy until conditions favour their activity. Likewise, when oxygen drops again, anaerobic processes resume rapidly. This adaptability ensures that hypoxic zones remain chemically active regardless of fluctuations. Microbial communities do not need stable conditions to function. They shift between different modes depending on what the water provides. The efficiency of microbial processes in hypoxic zones reveals why larger animals struggle to compete. In oxygen-rich waters, animals and microbes work together to recycle nutrients. In oxygen-poor regions, microbes take over nearly all of that responsibility. Their flexibility, combined with their minimal energy needs, allows them to dominate environments where oxygen-based life cannot persist. Their empires grow not through visible expansion but through biochemical influence. They shape the pH of the water, the distribution of nitrogen, the cycling of sulfur, and the transformation of carbon. Their presence defines the chemistry of the habitat more than any other group. These microbial communities also create conditions that further discourage the return of oxygen-dependent animals. Some of the byproducts they produce, such as hydrogen sulfide or ammonia, make the environment hostile even if oxygen returns temporarily. This does not mean that larger animals cannot re-enter the area, but it means that conditions may not support a full recovery without additional physical mixing or a reduction in nutrient input. The dominance of microbes can create a self-reinforcing cycle where their activity maintains the chemical state that benefits them, slowing the return of more oxygen-dependent organisms. In regions where hypoxia develops seasonally, microbial communities undergo recurring shifts. During periods when oxygen increases, some anaerobic processes slow down. When oxygen declines again, those same pathways re-emerge. Over time, these cycles select for microbes capable of rapid adjustment. The result is a stable but flexible community capable of functioning throughout the year. These patterns reveal the resilience of microbial empires. They adapt continually, responding not only to long-term changes, but also to short-term fluctuations in water chemistry. Understanding these microbial communities is essential for grasping how hypoxic zones develop and persist. Larger organisms may draw attention when they flee or die, but the underlying mechanics of the ecosystem depend on the microorganisms that remain. They are the ones that define the chemical environment, process the organic matter, and determine how nutrients move. Their activities shape the habitat in ways that affect everything around them. This microbial dominance marks a point where the ecosystem has shifted entirely away from the patterns common in oxygen-rich waters. As the oxygen continues to decline or remain absent, these microbial processes intensify, creating the conditions that define the next stages of hypoxia, where only organisms that can exploit the margins or drift through the boundaries continue to function within the evolving system. In the low oxygen layers of the ocean, where the water feels heavy and the usual rhythm of marine life fades, a particular group of creatures continues to move with surprising persistence. They drift along the boundary where oxygen begins to disappear, navigating a region that challenges almost every species that enters it. These organisms do not thrive in the same sense that animals in healthier waters do, but they persist, and their persistence reveals how life can function in narrow spaces where conditions change abruptly from tolerable to deadly. These drifters are creatures that rely on movement not powered by strength or speed, but by careful timing, efficient physiology, and the ability to exist in a zone that many other species avoid. One of the most recognizable drifters in low oxygen regions is the jellyfish. Jellies have bodies that depend very little on complex tissues, and this simplicity allows them to tolerate conditions that push other animals past their limits. Their movements require minimal energy. They pulse gently, propelling themselves forward in small increments, and when the water column stabilizes around them, they let the currents do most of the work. Their tissues absorb enough oxygen to maintain essential functions, and because their demands are low, they are able to continue feeding even in areas with very limited resources. The absence of competitors gives them an opportunity to occupy territory that was once crowded with fish. They consume small plankton or drifting organic particles, using a strategy that fits naturally within the slow pace required to survive at the edge of hypoxia. Some species of comb jellies also appear frequently in these regions. Unlike true jellyfish, comb jellies move using rows of cilia that beat in coordinated waves. These movements generate very little turbulence and require far less energy than muscular propulsion. Their transparent bodies blend into the water around them, and their feeding relies on sticky cells that capture plankton as they drift. In oxygen-poor layers, these animals take advantage of lower predation pressure. Many predators cannot enter these regions without risking oxygen deprivation. With fewer threats around, comb jellies drift more openly, expanding their feeding area and maintaining their slow but steady movements. Zooplankton represent another important group of drifters that frequent the edges of hypoxia. These small organisms include copepods, amphipods, krill, and various larvae of fish and invertebrates. In oxygen-rich waters, zooplankton engage in vertical migrations, travelling upward at night to feed and downward during the day to avoid predators. In hypoxic waters, these migrations become more complicated. Many zooplankton approach the low oxygen layer cautiously. Some remain just above the boundary, using it as a refuge where predators cannot follow. Even though they cannot stay in the hypoxic layer for long, their brief visits allow them to exploit resources unavailable in the layers above. Copepods in particular have shown an ability to enter and exit low oxygen waters in short intervals. Their movements are rapid but controlled. They swim down, gather what food they can, and return upward before oxygen levels drop below their tolerance threshold. The energy cost of these movements is significant, but the reduced predation pressure makes the strategy worthwhile. In healthy waters, copepods face threats from fish, jellyfish, and larger zooplankton. In hypoxic waters, many of these predators retreat. This gives copepods a temporary advantage. Their resilience lies not in the ability to withstand low oxygen, but in the ability to judge how long they can remain near its edge. Some krill species exhibit similar behaviour, though they generally avoid deep hypoxia due to their high oxygen demands. They often gather at the upper boundary, forming dense layers that track the thin band where oxygen begins to decline. These aggregations attract a few oxygen-tolerant predators that remain near the boundary, creating a narrow zone where life is compressed into a tight vertical space. Krill depend heavily on oxygen to power their constant swimming, and so they rarely enter deeper hypoxic layers, but their presence near the edge reveals how tightly the ecosystem connects to this boundary. Various larvae also drift along this narrow region. Larvae of fish, crustaceans, and mollusks often remain in higher oxygen waters, but during certain developmental stages some drift into lower layers. These larvae rely on small body size and lower metabolic demands to persist temporarily in conditions that would be difficult for adults. Their presence near the edge reflects both accidental drift and intentional use of a region where predators are less abundant. For some species, the boundary acts as a temporary refuge. For others, it becomes a risky passage they must survive as they grow. Drifting animals in these regions often rely on density differences in the water to maintain their positions. Many zooplankton possess oil droplets or gas-filled structures that allow them to regulate buoyancy. These adaptations generally assist them in moving between layers, but they also help them remain near the hypoxic boundary without sinking deeper. Some species actively avoid sinking by making small, rapid movements. Others use minimal burst of energy to correct their position whenever they drift too far downward. The balance between buoyancy and active control determines how long they can remain near the edge without entering lethal oxygen levels. Another type of drifter commonly found in low oxygen zones is the larvacean. These small animals build delicate mucus-based structures known as houses. These houses filter water, trapping food particles and channeling them toward the larvacean's feeding apparatus. When the house becomes clogged, the animal abandons it and builds a new one. In hypoxic waters, larvaceans alter the frequency with which they build new houses, conserving energy when food availability drops. Their houses also act as floating structures that keep them near specific layers within the water column. Because larvaceans rely on filtering water rather than active pursuit, they can continue feeding even when their environment becomes stressful. Their strategy depends on low energy expenditure paired with steady passive feeding, making them well suited to survive near the boundary of hypoxia. Some gelatinous zooplankton have internal adaptations that help them persist longer than expected. Certain species of salps and pyrosomes, which form long tube-shaped colonies, maintain slow but continuous water flow through their bodies. This flow brings oxygen to their tissues while also drawing in food particles. These colonies drift slowly with currents, and their low metabolic needs allow them to endure periods of reduced oxygen more effectively than many crustaceans or fish larvae. They do not move deliberately through low oxygen regions. Instead, they persist as passive drifters, adjusting their filtering and feeding strategies as the surrounding conditions change. Another drifter group consists of small pelagic worms known as ketognaths or arrow worms. These animals are ambush predators that drift patiently until prey swims close enough for them to strike. Their bodies require oxygen, but some species tolerate mild hypoxia for short periods. They remain near the upper boundary, where they can capture copepods that venture downward. By hunting in the narrow zone between oxygen-rich and oxygen-poor water, arrowworms exploit the concentration of prey without entering the most severe conditions. Their strategy depends on sudden bursts of activity followed by long intervals of drifting. This minimises their oxygen consumption and allows them to continue hunting near the edge. An interesting pattern in hypoxic regions is the temporary success of species typically outcompeted in healthier waters. When oxygen falls, dominant competitors retreat, leaving behind space and food for drifters that usually remain in the background. For example, some small medusae gain a significant advantage when fish leave the area. Their ability to drift and feed gently suits them well when the pressure from stronger competitors disappears. They are opportunistic in these conditions, not because they thrive in low oxygen, but because the absence of competitors gives them greater freedom to move and feed. Some fish species also behave as drifters under hypoxic conditions. These include small pelagic fish such as anchovies and sardines. Although they cannot survive within the hypoxic layer itself, they often gather at the boundary. When pushed upward by expanding hypoxic zones, these fish form dense schools near the surface, drifting slowly and reducing their activity. They rely on the limited oxygen available while waiting for conditions to improve. Their presence near the edge has large ecological effects, attracting predators that are capable of exploiting the concentrated schools. The predators themselves must remain near the surface, since diving deeper becomes difficult as oxygen drops. This compression of the food web brings interactions that would normally be spread across a vertical range into a very small space. Some crustaceans also use drifting strategies. A few species of mysid shrimp, for example, move through the boundary in small groups. Mysids are known for their sensitivity to environmental changes, yet some tolerate brief exposure to low oxygen by slowing their swimming and relying on rhythmic, minimal strokes of their appendages. They form small clusters that drift together, maintaining their position in the thin layer where oxygen remains. Their ability to feed on small particles and their flexible movement patterns make them effective drifters when conditions become challenging. Drifting at the edge of hypoxia is not without risk. For many species, the line between survivable and lethal conditions is extremely thin. A small change in temperature, salinity, or water movement can push them into oxygen levels that their bodies cannot tolerate. This sensitivity forces them to respond quickly. They must move upward or laterally when the oxygen boundary shifts. The speed of their response determines whether they survive. This challenge shapes the population structure of drifters. Those with faster reaction times or more efficient sensory systems persist, while others do not. Some organisms detect low oxygen through chemical receptors. They sense changes in the concentration of dissolved gases, allowing them to adjust their depth almost automatically. Others detect changes in pH or redox potential, which often correlate with oxygen levels. These sensory mechanisms are essential for navigating the shifting boundary. Without them, drifters would be unable to remain in the narrow space where survival is possible. Drifting species near the hypoxic layer also influence the movement of nutrients. As they feed, excrete waste, and interact with microbial communities, they transport nutrients upward into brighter, more oxygenated waters. This vertical exchange is subtle, but important. It provides small contributions to nutrient cycling that help support plankton populations above the hypoxic layer. Their movements also transport microbes or organic particles across boundaries, creating a slow but consistent transfer of material that shapes the chemical structure of the water column. Some drifters play roles in transferring carbon between layers. Zooplankton fecal pellets, for example, sink quickly through the water. In oxygen-rich areas, these pellets are consumed or broken down rapidly. In hypoxic zones, microbial activity slows. Pellets drift further downward without decomposing fully, delivering carbon to the sediments. This transfer supports microbial communities in deeper layers and influences the long-term build-up of organic matter in hypoxic basins. The presence of drifters ensures that carbon continues to move even when many large organisms have left. Because drifters rely on passive movement, their distribution often mirrors the structure of the water column. When the hypoxic layer expands, drifters shift with it. When the boundary rises or falls, so do they. These adjustments create dynamic patterns that vary over days, seasons, and years. Their populations may increase when conditions favour them, or decrease when food becomes scarce. In long-lasting hypoxic zones, some drifters develop populations specifically adapted to the boundary zone. These populations differ from those in healthier waters, suggesting that the edge of hypoxia acts as its own ecological niche. The boundary is also a region of intense competition among drifters themselves. Even though predation pressure is reduced, food resources remain limited. Drifters must balance their need for food with their need to avoid deeper layers. Those that linger too long in one area risk exhausting the available resources. Those that move too quickly risk sinking into dangerous conditions. Their strategies vary. Some rely on fast, repeated trips downward, followed by upward retreat. Others drift horizontally to locate new patches of plankton. In areas with strong stratification, drifters may remain confined to thin horizontal bands, creating high densities that lead to rapid resource depletion. These patterns highlight the delicate balance required to survive along the edge of hypoxia. The collective behaviour of drifters influences how predators approach these areas. Fish that tolerate mild hypoxia hover near the boundary, waiting for opportunities to feed on concentrated zooplankton. Certain species of jellyfish become both predators and prey within these layers. Larger jellies may feed on smaller drifters, while smaller drifters use the spacing of the jellies as temporary shelter. This interplay adds complexity to the boundary region, reinforcing how tightly food webs compress under low oxygen conditions. Some drifters rely on chemical defences to survive near the boundary. A few planktonic species release compounds that deter predators. In oxygen-rich waters, these defences play a role, but are balanced by predation and competition. In hypoxic regions, these chemical defences become even more important because movement options are limited. Drifters cannot escape quickly, so they depend on strategies that reduce the likelihood of being eaten. Their chemical defences contribute to the survival of populations that would otherwise struggle as they drift near the edge. The presence of drifters near the hypoxic boundary shapes the ecosystem's recovery potential. When oxygen returns to deeper layers due to storms, upwelling or circulation shifts, drifters are among the first to respond. They descend cautiously, exploring areas that were previously uninhabitable. Their movements can bring small amounts of organic matter into newly oxygenated layers, jump-starting microbial processes that resemble those in healthier waters. Their return helps rebuild small links in the food web, providing prey for species that re-enter the region. Their role is small compared to the contributions of fish or crustaceans, but in the early stages of recovery, every transfer of energy matters. Drifters occupy a region defined by instability. The boundary between oxygen-rich and oxygen-poor water moves frequently. It shifts with temperature, salinity, and external forces. Drifters must respond to these changes with precision, using minimal energy while maximizing opportunity. Their strategies reveal how life can persist even when the margin for survival is narrow. Their existence along the edge of hypoxia forms a transition between the slow-living creatures that remain in low oxygen and the microbial communities that dominate deeper, more extreme environments. As conditions continue to change, the drifters' movements influence how the ecosystem adjusts, setting the stage for the organisms that adapt to even more challenging conditions in the layers below. When oxygen declines to levels that squeeze nearly every form of marine life, a predictable shift begins to shape the organisms that remain. It is not a sudden transformation. It is gradual and built on countless small changes that accumulate over time. The animals that cannot tolerate the drop either abandon the region or die, leaving behind those that can endure the pressure. Among the survivors, certain patterns become noticeable. Many species become smaller, others slow their growth. Some adjust their body structures, shrinking their energy demands or redistributing the way they allocate resources. These changes are not merely responses to immediate stress, but long-term adjustments that reflect a broader effort to survive in a world where oxygen, food, and mobility are restricted. In the early stages of hypoxia, large animals are the first to leave. Their bodies require more oxygen, and they have greater energy needs. But once the decline reaches a sustained level, even some medium-sized animals begin to show reductions in body size over generations. Smaller bodies have lower oxygen requirements, shorter diffusion distances, and reduced metabolic demands. These traits become advantageous in low oxygen waters. In some regions where hypoxia recurs seasonally, fish populations show measurable reductions in average size. Their growth rates slow, and their maximum size decreases. Researchers studying these patterns have found correlations between prolonged low oxygen exposure and shifts in body proportions. These changes do not occur instantly. They happen gradually, driven partly by natural selection and partly by direct physiological constraints. Invertebrates show similar patterns. Some crustaceans shrink slightly, with individuals reaching maturity at smaller sizes than those living in well-oxygenated waters. Shrinking reduces the oxygen load required to maintain basic life functions. In these conditions, smaller individuals may survive where larger ones cannot. This does not necessarily make the population healthier, but it allows it to persist. The phenomenon resembles a narrowing of physical limits. Instead of growing until food or predation limits their size, these organisms grow until their oxygen environment restricts them. For many animals, shrinking is paired with a slowdown in growth. This slowing is not simply a matter of reduced food intake, it is rooted in diminished metabolic capacity. When oxygen availability is low, cells produce energy at lower rates. This reduced energy production affects everything from tissue development to movement. Growth becomes a slower process because the body cannot maintain the same pace of cell division. In some cases, animals delay reproduction until they reach a size that would normally have occurred much earlier in life. This delay can affect population dynamics, reducing the number of offspring produced over time. Some species employ physiological adjustments that help them manage their limited energy budgets. Many organisms reduce the number of active tissues in their bodies. For example, some fish shrink internal organs such as the liver or gills when oxygen is scarce. These organs may function at lower capacity during extended hypoxia, conserving energy at the expense of rapid metabolism. Certain bivalves reduce the size of their gonads or digestive glands under prolonged low oxygen, focusing their remaining energy on essential functions rather than reproduction or rapid digestion. These changes may seem severe, but they represent strategic choices shaped by survival pressures. In environments where oxygen limits nearly every biological process, organisms adjust their priorities. They maintain core functions such as breathing, circulation, and basic digestion, while reducing or suspending activities related to growth, reproduction, and movement. This prioritization reflects a hierarchy built into their physiology. When resources decline, survival overtakes expansion. Some species show more subtle adjustments, altering their behavior rather than their body structure. Many slow their movements drastically. This response is common among small crustaceans, worms, gastropods, and certain fish. Movement is one of the most energy-intensive activities for any organism. In hypoxic waters, where each movement increases the demand for oxygen, slowing down becomes a survival strategy. Instead of exploring or actively searching for food, animals remain still for long periods, moving only when absolutely necessary. In some systems, this slowing of movement becomes the dominant behavioral pattern. Animals that normally forage widely begin to feed locally. Predators that rely on active hunting shift to ambush strategies. Scavengers wait longer before moving toward new food sources. Even simple activities such as adjusting position in the sediment or rising toward the water column become calculated decisions rather than habitual behaviours. In the slow pace of hypoxic waters, the costs of movement often outweigh the benefits. Another key adjustment involves changing feeding habits. In well-oxygenated waters, many animals can afford to be selective. They pursue prey or graze actively. In hypoxic regions, choice becomes limited. Some species become less discriminating, eating lower quality food or relying on detritus more than they would under normal conditions. This shift supports survival, but at the cost of slower growth and reduced reproduction. The lower quality diet also contributes to the reduced body sizes common in hypoxic ecosystems. Some species alter their feeding times. Organisms that once fed during the day may switch to nighttime feeding, or vice versa, depending on subtle shifts in oxygen availability within the water. These adjustments reflect a flexibility that becomes crucial in areas where the distribution of oxygen changes rapidly. Even small fluctuations can determine whether a species successfully feeds or expends unnecessary energy. Many organisms also adapt by redistributing the amount of energy they devote to different stages of life. Some species produce fewer offspring under hypoxic conditions, but make each offspring slightly larger or more robust. This increases the likelihood that the offspring will survive in challenging conditions. Others produce more offspring but invest less energy in each individual. Both strategies reflect attempts to cope with unpredictable or stressful environments. Among microorganisms, shrinking and slowing take on different forms. Bacteria in hypoxic zones often reduce their cell size as part of a broader survival strategy. Smaller cells require less energy to maintain and allow faster diffusion of nutrients. In low-oxygen sediments, bacteria may divide less frequently, entering a slow growth mode that conserves resources. Although these bacteria may appear inactive, their biochemical reactions continue at reduced rates. Even in this slowed state, their role in nutrient cycles remains critical. Archaea, particularly methane-producing species, also exhibit slow growth adaptations. They can operate under extremely low energy conditions, performing chemical reactions that yield minimal energy. This allows them to survive in deep sediments where oxygen has been absent for long periods. Their growth is slow but steady, contributing to the long-term stability of these microbial communities. Planktonic organisms respond differently to shrinking pressures. Some plankton species reduce their body size during hypoxic seasons, producing smaller individuals that require less oxygen and can survive closer to the hypoxic boundary. These smaller forms drift more easily and have lower metabolic demands, giving them more flexibility to move through layers with varying oxygen concentrations. Their reduced size also minimises the risk of sinking into deeper, oxygen-poor waters. In a few cases, hypoxia triggers significant morphological changes. Certain bristle worms alter their gill structures, developing longer or more vascularized appendages to increase oxygen uptake. Some bivalves increase the surface area of their gills to extract more oxygen from the water. Fish may develop larger gill openings or increase the number of lamellae, improving the efficiency of their oxygen extraction. These changes can be costly. They require energy to develop and maintain. But in an environment where oxygen is the most limiting factor, these modifications provide valuable advantages. Some organisms adjust their internal chemistry to manage energy more effectively. They rely more heavily on anaerobic metabolic pathways, which do not require oxygen but produce lower amounts of energy. The buildup of byproducts such as lactate or succinate must eventually be cleared once oxygen returns, but the temporary shift protects the organism from immediate collapse. These chemical strategies help bridge the gap between oxygen availability and metabolic demand. The adaptations associated with shrinking and slowing also affect the roles of organisms within the food web. In oxygen-rich ecosystems, fast-moving predators and active grazers dominate. In hypoxic areas, these roles diminish. The organisms that persist occupy narrower niches. Their feeding patterns become more specialized. their interactions become slower and more predictable. Predators that rely on bursts of speed can no longer function effectively, leading to declines in predation pressure. This shift allows slow-moving prey species to survive even with limited defences. The entire system becomes defined by low-energy interactions. Another consequence of shrinking and slowing is the alteration of reproductive strategies. Some species reduce the frequency of reproduction. Others shift to producing eggs or larvae that can tolerate low oxygen. In certain fish species, hypoxia reduces egg size or changes the timing of spawning events. For invertebrates, reproductive cycles may delay or accelerate depending on seasonal patterns of oxygen availability. The variability in these strategies demonstrates how organisms adjust not only to their immediate conditions, but also to the patterns they anticipate in the environment. Shrinking and slowing also shape how organisms manage stress. In oxygen-rich waters, animals may use rapid movement to escape predators or unfavourable conditions. In hypoxic zones, escape becomes difficult or impossible. Instead, organisms invest more energy into tolerating stress. This may include adjustments to their cellular membranes, stress response proteins, or mechanisms that maintain internal chemical balance under low oxygen. These physiological shifts allow them to persist in conditions that many species could not withstand. Some organisms develop behaviours that reduce exposure to dangerous conditions. They move into microhabitats where oxygen concentrations are slightly higher, such as cracks in sediments, localised upwelling zones, or areas where slight water mixing occurs. These microhabitats offer temporary refuge, and organisms adjust their movement patterns to exploit them. They may remain in these pockets until oxygen declines, then shift to another location when conditions permit. This approach allows them to navigate hypoxic waters without exceeding their metabolic limits. Among mobile species, one of the most notable adaptations is the shift from active pursuit to passive feeding. Instead of chasing prey, organisms rely on drift, debris, or sediment-bound resources. This reduces energy expenditure and aligns feeding strategies with the diminished availability of oxygen. Passive feeding strategies become especially important for species living near the edges of hypoxic zones, where movement in either direction must be carefully controlled. Slow-living organisms also influence how materials move through the ecosystem. Their reduced feeding and movement change the distribution of organic matter. Sediments accumulate more partially decomposed material due to lower rates of consumption. Microbial activity takes over most decomposition, but at a slower pace. This altered nutrient cycling affects every layer of the ecosystem, reinforcing the conditions that favour slow and small organisms. In some systems, these adaptations drive long-term changes in population structure. Certain species become more abundant because they are well suited to hypoxia. Others decline because they cannot adjust quickly enough. The changes are not uniform. Some populations show fluctuations depending on the duration and severity of hypoxia. Others stabilise as they adapt fully to low oxygen conditions. Over time, the community composition shifts toward organisms that specialise in slow, low-energy living. These patterns also highlight the importance of physiological flexibility. Species that can adjust multiple aspects of their biology, body size, metabolism, behaviour, and reproduction, have a greater chance of persisting. Flexibility becomes more valuable than any single adaptation. The organisms that survive are those that can change quickly when conditions shift and endure long periods of resource limitations. Understanding how life shrinks and slows in hypoxic regions shows how closely survival is tied to energy management. Every adaptation reflects a negotiation between what the organism needs to function and what the environment can provide. These negotiations shape everything from body structure to feeding habits. They create ecosystems where the pace of life is defined by scarcity, but where persistence continues. As conditions push organisms to their limits, these adjustments reveal the pathways that allow life to endure and prepare the foundation for even more specialized strategies in deeper, more extreme areas where oxygen becomes nearly absent. and chemical survival takes centre stage. When oxygen begins to disappear from the water and entire communities of organisms slow, shrink, or retreat, the transformation unfolding beneath the surface is more than biological. It is chemical, shaped by reactions that shift with every change in temperature, nutrient load, and microbial activity. A sea falling asleep is not a quiet process. It is a slow rearrangement of dissolved gases, ions, and compounds that determine what can live, what cannot, and how the system will behave as hypoxia deepens. To understand this quieter phase of the ocean's life, it helps to picture how each chemical step creates the next, building a progression that eventually leads to conditions far removed from what most marine animals consider normal. The first major chemical shift begins with a decline in dissolved oxygen. Under healthy conditions, oxygen enters the ocean from the atmosphere and from photosynthesis by phytoplankton near the surface. It moves downward through mixing and circulation. When the surface becomes warmer due to seasonal changes or long-term climate trends, that mixing slows. Warm water forms a stable layer that resists blending with the colder water below. This separation marks the start of chemical stratification, where different layers of water develop their own identities. The top layer carries oxygen but does not easily share it. The deeper layers consume what oxygen they have, but without mixing they cannot replenish it. As oxygen declines, the deeper layer becomes chemically isolated. Organisms living there continue to respire, consuming oxygen and releasing carbon dioxide. Microbes break down sinking organic matter, using oxygen during the process. Because there is no fresh supply coming from above, the oxygen concentration decreases steadily. Once it drops below a certain threshold, the chemical reactions that relied on oxygen begin to slow, and alternative pathways become more important. One of the earliest chemical changes involves the accumulation of carbon dioxide. When oxygen is lost from water, carbon dioxide does not simply disappear. It dissolves into the water and reacts with it to form carbonic acid. This lowers the pH, making the water slightly more acidic. Even small changes in pH influence chemical processes. Certain minerals dissolve more readily. Nitrogen compounds shift into different forms. Animals that depend on stable pH for maintaining internal balance become stressed. In healthy oceans, these changes remain moderate. In hypoxic waters, however, acidity can rise much higher, especially in isolated basins or areas with large amounts of decaying organic matter. The decline in oxygen changes the way nitrogen moves through the ecosystem. Nitrogen is essential for life, and its chemical transformations determine how much is available for algae and other organisms. In oxygen-rich waters, bacteria convert ammonia into nitrite, and then into nitrate through a process known as nitrification. This process requires oxygen. When oxygen levels drop, nitrification slows or stops. The bacteria that rely on oxygen begin to decline, and other microorganisms take over, shifting nitrogen into different forms. One of the most important of these processes is denitrification, where nitrate is converted into nitrogen gas. This reaction removes nitrogen from the water entirely, decreasing the amount available for algal growth. At first glance, this might seem like a positive effect. Less nitrogen means fewer algal blooms, but in reality the chemistry is not that simple. Hypoxia often develops in areas with high nutrient loads from land, and the pace at which nitrogen is removed rarely matches the pace at which it enters. Furthermore, denitrification produces small amounts of nitrous oxide, a greenhouse gas far more potent than carbon dioxide. Although only trace amounts escape into the atmosphere, their contribution becomes part of the broader chemical footprint of hypoxic zones. As oxygen levels continue to drop, another set of chemical pathways becomes dominant. Some bacteria use nitrate as a substitute for oxygen. When nitrate becomes limited, these microbes turn to other compounds such as sulfate. Sulfate reduction is one of the most significant chemical reactions in hypoxic and anoxic zones. Sulfate-reducing bacteria convert sulfate into hydrogen sulfide, a gas known for its sharp odour and strong toxicity to many organisms. Hydrogen sulfide accumulates in sediments, and sometimes in the water itself. In areas where water remains stagnant, the gas can build up to concentrations capable of harming fish and invertebrates if mixing suddenly brings it upward. Hydrogen sulfide does not always remain in its reduced form. In layers above the sulfate-reducing zone, another group of microbes oxidizes the hydrogen sulfide, converting it back into sulfate. These opposing reactions form a chemical gradient that defines the middle layers of hypoxic regions, where hydrogen sulfide and small amounts of oxygen or nitrate meet. Sulfur-oxidizing bacteria flourish. Their activity produces visible mats along the seafloor or suspended layers in the water column. These mats indicate the locations where chemical reactions are most intense. Their presence is a reminder that even though the sea appears inactive, its chemistry remains in constant motion. Another consequence of low oxygen is the accumulation of ammonium. When organic matter breaks down under anoxic conditions, nitrogen is released primarily in the form of ammonium rather than nitrate or nitrite. Ammonium can build up in the deeper layers of hypoxic basins, especially in areas with significant sediment inputs or high primary productivity. This ammonium-rich layer becomes a reservoir of reduced nitrogen that remains trapped until mixing or upwelling brings it closer to the surface. When this happens, a sudden influx of ammonium into oxygenated waters can trigger intense microbial activity or fuel new algal blooms. Iron and manganese also undergo significant changes in chemistry as hypoxia develops. Under oxygen-rich conditions, these metals form solid oxides that settle into sediments. When oxygen disappears, the metal oxides dissolve, releasing dissolved iron and manganese into the water. These reduced metals can diffuse upward through the water column. When they reach oxygenated layers, they reoxidize and form solid particles again. This repeated cycle creates a dynamic movement of metals within hypoxic basins. The accumulation of reduced iron or manganese can alter nutrient availability, stabilize or destabilize sediments, and influence how other chemicals move. Another chemical behavior seen in hypoxic waters is the reduction of phosphate. Under normal conditions, phosphate binds to iron oxides in sediments. But when oxygen disappears and iron dissolves, phosphate is released into the water. This can create a feedback loop. The phosphate fuels algal growth in surface waters. When the algae die and sink, they further deplete oxygen as microbes break them down. The cycle continues with each new influx of nutrients, making hypoxia more severe and more persistent. Deep within the sediments of hypoxic zones, methane production becomes one of the last remaining metabolic pathways. When oxygen, nitrate, sulfate, and other compounds have been used up, certain archaea begin converting organic matter into methane. This process is slow but continuous. Methane accumulates in the deepest layers of sediment, forming pockets of gas that remain trapped unless disturbed. Some methane escapes upward, where methane oxidizing bacteria consume it in a thin layer above the sediments. This interaction creates another chemical boundary, completing the progression of reactions that occur as oxygen disappears. Chemical stratification influences how energy moves through the system. In oxygen-rich environments, energy flows through organisms that consume food and release carbon dioxide. In hypoxic waters, energy moves mostly through chemical gradients. Microbes use compounds created by other microbes. Chemical reactions unfold in layers that depend on the availability of specific ions or molecules. Each layer reflects the condition of the water at that depth. The chemistry becomes a map of the habitat, revealing which organisms can survive and which processes dominate. This separation of layers has implications for larger-scale ocean processes. In some regions, hypoxic waters produce localized acidification due to high carbon dioxide concentrations. This acidification can weaken shells or interfere with the ability of some species to maintain internal chemical balance. In deeper waters, the buildup of ammonium, hydrogen sulfide, and methane creates conditions that push many organisms beyond their tolerance limits. Even when oxygen returns, these residual chemicals can delay recovery. In areas where hypoxia develops seasonally, the chemistry cycles between oxygen-rich and oxygen-poor states. During periods of high oxygen, nitrification resumes, pH rises slightly, and metal oxides reform. During low oxygen, these processes reverse. Over many cycles, this repetition changes the stable chemistry of sediments and the water column. Sediments in these regions may become more enriched with reduced compounds. The water becomes more prone to chemical swings that affect the organisms living there. Temperature influences this chemistry as well. Warmer waters hold less oxygen and accelerate microbial metabolism, increasing the rate at which oxygen disappears. Warmer temperatures also speed up chemical reactions involving nitrogen and sulfur. In some cases, heat causes stratification to tighten, making it harder for oxygenated surface water to mix with deeper layers. The combined effect amplifies hypoxia and strengthens the chemical gradients that define these waters. Nutrient input from land also plays a critical role. Rivers carry nitrogen, phosphorus, and organic carbon from agricultural and urban areas. These nutrients fuel algal blooms, which eventually die and sink. The increase in organic matter boosts microbial respiration and accelerates the consumption of oxygen. Hypoxic zones in coastal regions often correspond to areas with high nutrient runoff. Their chemistry reflects this influence, exhibiting elevated nitrate, ammonium, phosphate, and organic carbon concentrations. Salinity shapes chemical behavior as well. Fresh water entering the ocean from rivers is lighter than seawater. It forms a surface layer that increases stratification. The reduced mixing allows oxygen to remain near the surface, while the deeper layers become isolated. Salinity also affects the solubility of certain compounds, including gases and metals. These interactions contribute to the chemical structure of hypoxic regions. Circulation patterns determine where hypoxia develops and how long it persists. In some regions, currents push oxygen-poor water into deeper basins where it remains trapped. In others, upwelling brings nutrient-rich but oxygen-poor water to the surface, affecting local ecosystems. The movement of water influences how chemical layers form, how long they last, and how they shift over time. The chemistry of a sea falling asleep not only shapes the present, but also influences the potential pathways for recovery. When oxygen returns, either through mixing or circulation changes, the chemical gradients collapse. Hydrogen sulfide may become oxidized. Iron and manganese may form solid oxides again. Ammonium may convert to nitrate. But these changes do not happen instantly. The rate at which the chemistry rebalances influences how quickly biological communities can return. In some cases, the chemical legacy of hypoxia persists long after oxygen levels increase. For organisms living in these regions, the chemistry defines their world. Microbes rely on chemical gradients for energy. Worms and other benthic organisms navigate layers where compounds shift from supportive to harmful. Drifters near the boundary encounter changes in pH, ammonium, or metal concentrations. Certain species sense hydrogen sulfide or lowered pH as warnings and retreat. Others tolerate these changes or use them as cues to locate microhabitats where conditions remain stable for short periods. The complexity of these chemical transitions reveals the interconnectedness of ocean processes. Each reaction builds on the one before it. The decline in oxygen triggers changes in nitrogen. Those changes affect sulfur. Sulfur reactions influence metal behavior. Metal behavior affects phosphate cycling. Together, these processes reshape the environment in ways that influence both the organisms that remain and the ones that may eventually return. Understanding these chemical transformations helps explain why hypoxic zones can be difficult to reverse. Once the system begins to operate on chemical pathways that do not require oxygen, restoring oxygen alone is not enough. the accumulated compounds must be rebalanced. Microbial communities must adjust. Sediments must shift to new chemical states. These changes take time, and their pace depends on the scale and duration of the hypoxic event. As these transformations continue, the sea becomes defined more by chemical interactions than by biological ones. The slower pace of life, the shrinking of organisms, and the rise of microbial processes reflect a world in transition. The chemistry sets the stage for the next developments, influencing how the ecosystem evolves when the oxygen is pushed to its limit and the layers below begin to resemble environments once found only in small, isolated pockets of the ocean. When an ocean region settles into hypoxia, the entire system becomes defined by stillness. The deeper layers hold their chemical gradients without interruption. Microbial communities operate steadily. Slow-living animals conserve energy and remain near pockets of slightly better conditions. Above them, the surface may look normal, yet beneath it, the water has settled into a pattern that resists change. What breaks this pattern are the physical forces capable of disturbing the stratification that keeps oxygen out and keeps reduced chemical compounds trapped below. Storms and currents play that role, shifting the balance in ways that can temporarily relieve or greatly intensify the conditions below. Their impact depends on timing, strength, and the structure of the water column before they arrive. One of the main reasons hypoxia persists is stratification. Warm water sits on top, lighter than the cooler water beneath. This layering prevents the downward mixing of oxygen. When storms or dominant wind patterns disturb the surface, they can force the warm layer to churn. If the churning is strong enough, some of the surface oxygen mixes into deeper layers. This is one of the simplest pathways towards short-term relief. However, not every storm produces enough turbulence. Small storms may only disturb the uppermost water and fail to reach the deeper layers where oxygen is needed. The effect depends on how much energy the wind transfers into the water and how tightly the layers have separated. In some systems, storms create vertical mixing that reaches shallow hypoxic layers. Coastal regions, for example, often experience quick but shallow stratification. A strong storm may break through the boundary between surface and bottom waters, pushing oxygen downward. When this happens, the oxygen-starved animals on the seafloor respond rapidly. Crabs begin moving again. Shrimp return to open areas. Fish re-enter the region if the oxygen pulse is strong enough. Microbial processes adjust as well. Nitrifying bacteria begin converting ammonia and nitrite into nitrate again. Sulfate-reducing bacteria slow their activity when oxygen penetrates the sediment. Over time, these reactions reshape the chemistry, at least temporarily. Yet storms do not always bring relief. In some circumstances, storms transport large quantities of organic material from rivers or disturbed sediments into coastal waters. This material fuels microbial decomposition. When microbial demand increases, oxygen consumption rises. If the water is not mixed deeply enough, this sudden addition of decomposing matter can worsen hypoxia. In other words, storms that stir up sediment or increase nutrient inputs can deepen the problem instead of easing it. This dual role makes storms unpredictable. Their influence depends as much on what they bring into the system as on how strongly they mix it. Another important factor is the timing of storms. Early season storms, arriving before the peak of summer stratification, can prevent hypoxia from developing fully. They disrupt the surface layer, breaking down the temperature gradient before it becomes too strong. This early mixing distributes nutrients and oxygen more evenly. By contrast, late-season storms may arrive when hypoxia is already extensive. In these cases, the mixing may serve as a short-lived relief, but the deeper chemical conditions may persist and react strongly to the influx of oxygen. One of the more dramatic consequences of mixing involves hydrogen sulfide. In some regions where hypoxia has persisted for long periods, Hydrogen sulfide accumulates in deep waters or in sediment layers. If a powerful storm mixes water too rapidly, pockets of hydrogen sulfide may rise toward the surface. This can be dangerous for marine life. Even small increases in hydrogen sulfide can cause fish kills. The gas interferes with important cellular processes, and the fish that encounter it often suffer immediate effects. Because hydrogen sulfide is produced in deeper layers, storms strong enough to lift it upward can convert a chemically dormant hazard into an active threat. Currents influence hypoxia differently. While storms act through turbulence and short-term disturbance, currents work through large-scale movement of water across vast distances. Ocean currents transport warm and cold water, nutrient-rich and nutrient-poor water, and oxygenated and oxygen-depleted water. The direction, strength, and variability of currents determine where hypoxic zones form, how they shift, and whether they intensify or dissipate. In some coastal regions, currents push oxygen-poor water toward the shore. This often occurs in areas downstream from large river systems where nutrient loads are high. When the current brings this water into shallower regions with clear stratification, the hypoxic zone expands. Animals living near the bottom suffer first. They migrate if they can. Those unable to move endure the declining conditions until their tolerance is exceeded. Microbes already active in the sediments respond quickly, accelerating processes that favour low oxygen chemical pathways. In other areas, currents bring oxygen-rich water into hypoxic zones. This occurs when deeper ocean currents rise along continental shelves or enter basins where oxygen has been depleted. The influx of fresher, cooler, oxygenated water can collapse existing chemical gradients. Once oxygen makes contact with hydrogen sulfide in the water column, sulfur compounds oxidize. Iron and manganese begin to precipitate again. Ammonium may start converting into nitrate. These changes shift the microbial composition. Sulfate-reducing bacteria retreat deeper into sediments. Denitrifiers slow their activity. The reintroduction of oxygen also allows animals to return, though often only briefly if the mixing does not continue. The dynamic interaction between currents and local stratification can create complex patterns. A current may travel along a coast and only partially mix with bottom waters, creating layers where oxygen varies dramatically over short distances. These shifts may produce narrow vertical windows where fish and crustaceans cluster. In these thin layers, life compresses as animals crowd into the oxygenated band, avoiding both the low oxygen region below and the predator-rich waters above. Their movements reveal how tightly conditions are linked to the physical flow of water. In regions where upwelling dominates, oxygen-poor but nutrient-rich deep waters rise to the surface. This is a natural phenomenon and supports productive ecosystems in many parts of the world. But upwelling can also intensify hypoxia when the rising water contains very low oxygen levels. The nutrients in the upwelled water fuel large phytoplankton blooms. When these blooms die, the sinking material increases microbial decomposition, drawing oxygen out of the water. This combination of upwelling and nutrient load can produce recurring hypoxic zones that wax and wane with seasonal wind patterns. In some areas, currents shift hypoxic zones laterally. Rather than mixing them vertically, they move them horizontally along coastlines or across basins. This movement affects every organism living in the region. Fish often encounter hypoxic water unexpectedly. They may move into the region temporarily and then retreat when oxygen declines. In extreme cases, currents can push hypoxic water into estuaries, lagoons, or enclosed bays where mixing is limited. Once trapped, the hypoxic layer intensifies and remains until circulation patterns shift again. The pairing of storms and currents creates additional complexity. After a storm passes, altered wind patterns may redirect currents, moving newly mixed water into different areas. Strong storm surges can push water offshore, drawing deeper water upward in its place. The interaction between these processes determines whether the aftermath of a storm brings recovery or exacerbation. Each system responds differently depending on the shape of the coastline, the presence of river inflows, the internal structure of the water column, and the timing of seasonal patterns. A significant phenomenon associated with storms is the displacement of surface waters. Hurricanes and cyclones produce immense vertical mixing. Their power can bring oxygen into deep layers that rarely receive it. They also disrupt stratification on a large scale, potentially reversing months of separation in a matter of hours. The sudden introduction of oxygen into deep hypoxic layers triggers rapid changes in microbial activity. Aerobic microbes become active again. Anaerobic microbes retreat or adjust. Chemical reactions involving nitrogen, sulfur, and iron undergo abrupt shifts. This reset can provide temporary relief for benthic communities. However, storms also generate massive amounts of runoff from land. Rivers swell with rainwater, carrying nutrients, sediments, and organic matter into coastal waters. The sudden increase in nutrient input can lead to algal blooms once the storm passes. When those blooms die, decomposition may consume more oxygen than the storm introduced. This delayed effect creates a pattern where the storm provides short-term mixing followed by long-term decline. The severity of the hypoxia that follows depends on how much material entered the ocean, how quickly microbial communities process it, and whether subsequent mixing events occur. In deeper ocean basins where hypoxia may develop without direct nutrient inputs from land, currents play a more significant role than storms. Some basins remain isolated for long periods due to circulation barriers. In these environments, deep currents may occasionally intrude, bringing oxygenated water into areas that have been stagnant for years. When this occurs, the chemical gradients collapse quickly. Hydrogen sulfide may be oxidized. Ammonium may be converted to nitrate. Methane near the bottom may be consumed by methanotrophic bacteria. These intrusions reset the system, but do not always last. If the basin remains poorly connected to larger currents, the oxygen introduced may be consumed quickly, and hypoxia can return. The hopeful stirring of waters that comes from storms and currents is not only physical, but ecological. Once oxygen begins to return, even temporarily, animals respond. Fish may re-enter the area, testing the conditions with cautious movements. Some species hover near the boundary, waiting to see if oxygen persists. Others begin feeding again, using the opportunity to rebuild energy reserves depleted during hypoxia. Crustaceans leave sheltered areas and return to open feeding grounds. Worms and bivalves resume normal activity in sediments. These responses occur rapidly, because the organisms living near hypoxic zones have adapted to exploit any improvement in conditions. Drifters near the boundary also adjust their behaviour. Zooplankton, that had been restricted to a narrow layer, may explore deeper depths. Their movement helps transfer nutrients upward and downward, influencing microbial activity. Jellyfish may shift their vertical positions as well, following the changing structure of the water column. These subtle changes in movement contribute to the redistribution of energy within the system. For microbial communities, the return of oxygen shifts the dominant chemical pathways. Aerobic respiration becomes possible again. Nitrification resumes, allowing nitrogen to move back into its oxidized forms. Iron and manganese oxidize, altering sediment chemistry. These changes help stabilize the environment, at least in the short term. Although the full recovery of a hypoxic zone requires more than a single mixing event, these steps contribute to a gradual restoration of balance. Currents and storms also influence long-term patterns of hypoxia. Regions with strong seasonal storms may experience hypoxia that fluctuates, but never reaches extreme levels. Areas with weak currents or limited storm activity may develop persistent hypoxic zones that expand over time. The patterns of these disturbances determine which species adapt, which persist, and which decline. Over many years, communities in frequently disturbed regions may evolve strategies that allow rapid exploitation of short-lived improvements. In contrast, communities in stable hypoxic zones adapt to long-term oxygen scarcity. Another important aspect of these disturbances is their role in transporting larvae and juvenile organisms. Some storms redistribute larvae from oxygen-rich waters into hypoxic zones. If the conditions improve long enough, these larvae may settle and grow. This can introduce new genetic diversity or shift local population dynamics. Currents also transport larvae across boundaries, influencing which species can recolonize areas recovering from hypoxia. The stirring of waters affects more than the immediate region. When storms or currents mix the water column, they transfer nutrients and oxygen across boundaries. This can influence primary productivity in nearby areas. Plankton respond to these changes, creating new feeding opportunities for small fish. Those fish attract to larger predators. The cascade of effects spreads outward, connecting hypoxic regions to healthier ecosystems. Even though hypoxia restricts much of marine life, the ocean retains mechanisms for renewal. The disturbances that mix water, redistribute nutrients, and shift chemical gradients provide windows of opportunity. These windows do not guarantee lasting improvement, but they allow the system to breathe, sometimes literally. They offer moments when the chemical and physical barriers that define hypoxic zones loosen. In those moments, organisms that have adapted to the margins of survival test the boundaries, respond to change, and begin the slow work of reclaiming the space. These interactions between physical and chemical processes set the stage for the next transitions. As currents shift again, or storms pass, the boundaries of hypoxia move. Some animals advance into recovering territory, others retreat. Microbial communities adjust their activity levels. The chemical environment reconfigures itself based on new inputs. This dynamic movement between decline and renewal continues to influence the trajectory of hypoxic zones, shaping the conditions that determine what comes next, as the system shifts again under the pressure of long-term change. When hypoxia first forms in any region of the ocean, its boundaries seem clear. Scientists can trace the depth at which oxygen drops below a critical threshold, map the horizontal spread, and observe which organisms flee or remain. Yet those boundaries are never permanent. Dead zones shift through time, not because they behave like living creatures, but because the physical and chemical forces shaping them keep changing. Storms, currents, shifting nutrient loads, seasonal temperature cycles, and long-term climate patterns all influence where low oxygen conditions appear. Over months, years, or decades, a dead zone can grow, shrink, move laterally, or vanish for a period before returning again. This movement is not accidental. It reflects the dynamic balance between what the water receives, what the organisms demand, and how the environment processes the inputs. One of the first observations made about shifting dead zones came from coastal areas influenced by large rivers. These rivers carry nutrients, sediments, and organic matter from land to sea. When the nutrient load increases, phytoplankton blooms occur. When those blooms die and sink, bacteria consume the organic matter using oxygen in the process. If stratification is strong enough to prevent mixing, the oxygen disappears from the bottom waters. Once hypoxia forms, the dead zone expands outward from the region, with the highest organic load. The movement looks like a spreading stain in the water column, but the spreading does not continue evenly. Instead, it follows patterns defined by currents, wind direction, coastal shape, and the depth of the continental shelf. Over time, researchers noticed that these zones rarely remained in the same place from year to year. They might appear closer to shore one summer, then farther offshore the next. The expansion may be wide during one season, and narrower in another. These changes reflect shifts in winds that alter the surface layer, changes in the river's nutrient load, or variations in temperature that affect stratification. Even slight differences in the timing of spring warming can shift the location of the boundary between oxygen-rich and oxygen-poor waters. When the warming begins early, stratification sets in sooner, giving more time for hypoxia to form. When the warming is delayed, storms may mix the water longer into the season, limiting hypoxia's growth. The timing alone alters the location and size of the dead zone. Another movement pattern emerges when hypoxic water is pushed by coastal currents. These currents travel along shorelines, carrying water masses from one region to another. When hypoxic water forms near a river mouth, the currents may transport it down the coastline. In some regions, the movement is slow, shifting the hypoxic layer a few kilometers over several weeks. In others, the movement is rapid. Within days, an area that had decent oxygen levels can find itself enveloped in water that is nearly depleted. The organisms living there encounter hypoxia suddenly and respond by fleeing if they can. The fish that once swam comfortably along the seafloor rise toward the surface. Crabs crawl into shallower waters. Some organisms cannot escape, and their presence becomes part of the accumulating organic matter that worsens hypoxia. This travelling pattern occurs in several parts of the world. The shifting hypoxia is not due to internal growth, but to the physical movement of the water itself. Because the water mass travels, the dead zone appears to migrate. What looks like an expansion is sometimes just relocation driven by circulation patterns. These movements allow hypoxic zones to impact areas that would otherwise remain unaffected. They also complicate predictions, since the movement depends on weather patterns, seasonal shifts, and long-term oceanic trends. Another factor influencing this movement is the presence of eddies. Eddies are circular water masses that trap water inside their rotating structure. When an eddy forms near a hypoxic region, it can gather oxygen-depleted water and carry it a considerable distance. The trapped water rotates within the eddy, remaining stratified and gradually deepening the hypoxia inside it. As the eddy drifts, it transports the hypoxic water far from where it originated. In some cases, eddies carry dead zone water into open ocean regions where it eventually mixes and dissipates. In other cases, they deliver it into semi-enclosed bays or coastal pockets where circulation is weak. Once trapped, the water may remain hypoxic for extended periods, expanding the dead zone in areas otherwise less vulnerable. Dead zones also grow because the internal processes that support hypoxia feed back into themselves. When organic matter accumulates, microbial decomposition increases. This increases oxygen consumption. When oxygen vanishes, chemical changes in the sediment release nutrients like phosphate and ammonium. These nutrients then fuel new algal growth in surface waters if they reach the upper layer. As those algae die, they sink and continue to fuel oxygen consumption. This positive feedback increases the size of the hypoxic zone even if external nutrient inputs do not increase. The system begins to sustain itself through internal recycling. As long as stratification persists and circulation cannot break the cycle, the dead zone may grow through this internal mechanism. However, the growth rarely continues indefinitely. Seasonal processes bring interruptions. When temperatures drop in autumn or winter, stratification weakens. Cooler surface water gains density and begins to sink. This sinking forces deeper water upward, mixing the layers. The mixing introduces oxygen into the hypoxic region, shrinking or eliminating the dead zone temporarily. During these months, animals may return to the affected area. Crabs, fish, and shrimp recolonize the seafloor. Microbial processes shift back toward oxygen-based pathways. The chemical gradients collapse. But the relief is often temporary. When the next cycle of stratification begins, conditions worsen again. The return of dead zones follows a seasonal rhythm in many regions. Each summer, the dead zone regrows, sometimes reaching the same size as before and sometimes exceeding it. The variation depends on how the winter mixing unfolded, how much nutrient input the region received in spring, how quickly stratification formed, and how storms shaped early season conditions. Some years bring milder dead zones because storms mix the water unexpectedly. Other years bring larger dead zones because the nutrient input was high, or the stratification became intense early. In some regions, the return of hypoxia is not seasonal, but cyclical over longer periods. For example, certain semi-enclosed seas have experienced hypoxia that persisted for years, disappeared for a few decades, and then returned. These shifts correlate with long-term climate patterns, changes in river discharge, alterations in regional circulation, or human impacts on nutrient input. The cycles reflect large-scale environmental changes that shape the baseline conditions for hypoxia. When conditions favour stratification and high productivity, dead zones grow. When conditions shift toward stronger circulation or lower nutrient input, dead zones shrink or vanish. These cycles can last for decades, giving the appearance that the dead zone slept for a period before awakening again. Another long-term pattern involves the expansion of dead zones into deeper waters. Historically, hypoxia was often a coastal phenomenon driven by nutrient runoff. But as the ocean warms and stratification strengthens globally, deeper regions begin to experience oxygen declines. These deeper dead zones move slowly because the physical changes affecting them occur over large spatial scales. The decline in oxygen spreads from one layer to another as circulation weakens or deep water formation declines. Over time, the region of low oxygen expands vertically and horizontally. These deeper expansions do not appear or disappear with seasons but evolve gradually. Their movement reflects long-term shifts in global circulation patterns and climate-driven changes in ocean structure. Similarly, some dead zones return due to changes in agricultural and urban practices. When nutrient loads decrease as a result of policy changes or improved management, hypoxia may diminish. But if the nutrient loads later increase, the dead zones may return just as strongly or even more intensely. This return does not always require the same level of nutrient input that originally caused the hypoxia. Sediments enriched with decades of nutrient buildup can continue fueling hypoxia even after external inputs decline. This memory effect causes dead zones to reappear more readily, because the internal conditions support rapid oxygen depletion. In some estuaries, dead zones shift position due to tidal influence. During high tides, oxygenated seawater may enter, pushing hypoxic water farther upstream or downstream. During low tides, the weakened influence of seawater may allow the hypoxic water to expand back over the region it left. This pulsing movement creates a repeating pattern, where the dead zone shifts with tidal cycles. Organisms caught in these areas must tolerate rapid changes in oxygen availability. Those that cannot adapt leave the region or die. This tidal shifting builds a complex pattern of movement that differs from the long seasonal cycles observed in larger systems. Another important pattern occurs when dead zones spread laterally into regions where the topography traps them. Submarine canyons, coastal depressions, and basin-shaped areas create natural holding spaces for hypoxic water. When currents push low oxygen water into these geomorphological features, it becomes difficult for the water to exit. Once trapped, the region becomes prone to long-term hypoxia. Even minor mixing events may not be strong enough to move the water out. Instead, the dead zone persists, expanding slightly or contracting marginally, depending on the inputs and physical conditions. Over time, these areas become known for recurring or persistent hypoxia. Dead zones also move vertically. As surface warming increases or freshwater inflow strengthens stratification, the depth at which oxygen disappears rises. The hypoxic boundary moves upward, reducing the habitable space for benthic and pelagic organisms. Fish that once swam deeper shift to midwater levels. Zooplankton adjust their vertical migrations, avoiding sinking into the hypoxic layer. The upward movement compresses life into a narrower band, increasing competition and predation pressure near the boundary. This vertical compression plays an important role in shaping the ecosystem, often causing sudden declines in species that rely on deep habitats. In some systems, dead zones show a pattern of lateral movement followed by fragmentation. As currents push hypoxic water along the coast, patches of low oxygen may separate from the main body. These patches form isolated pockets where the oxygen decline continues. Meanwhile, the main dead zone moves on. These smaller pockets may dissipate quickly if they mix with surrounding waters, but in certain cases they persist, forming new centres of hypoxia. These satellite dead zones later merge with the main zone when conditions align. This merging and splitting create a pattern where the boundaries shift constantly, with hypoxia appearing in places that were clear only days earlier. Returning dead zones create new cycles of adaptation for organisms. When the hypoxia disappears briefly, animals recolonize the region. They lay eggs, feed, and establish territories. When the hypoxia returns, these animals must flee again or face declining conditions. This cycle favours species with rapid development, flexible movement patterns, and tolerance for environmental stress. Species that reproduce slowly or cannot move easily suffer declines. Over years, the recurring cycles reshape the biological community. The species best suited to the moving boundaries of hypoxia remain, while others disappear from the region. Dead zones also influence fisheries. When low oxygen waters move into areas used by fishers, catch rates decline. Fish retreat to higher oxygen areas, often closer to the surface or farther offshore. These shifts can disrupt local economies and create unpredictable patterns in fish availability. When the dead zone shrinks or moves away, fish return, but their behaviour may have changed due to prolonged stress. These patterns show how the movement of hypoxia affects not only the ecosystem but human communities as well. Because dead zones grow, move, shrink and return with such complexity, predicting their behaviour remains a challenge. Scientists use models that incorporate wind patterns, nutrient loads, temperature, salinity, circulation, and topography. Even with these tools, unexpected shifts occur. A single storm can disrupt months of stratification. A sudden change in current direction can expand the hypoxic boundary. A warm year or a heavy rainfall season can increase the severity dramatically. Understanding these shifting patterns is essential for anticipating how hypoxia will shape the next stages of environmental change. As the boundaries continue to evolve, the organisms that depend on the region must adapt repeatedly. The dead zones that move, grow and return create a landscape defined not by stability, but by ongoing transformation. Their shifting outlines influence how the chemical gradients develop, how the microbial communities adjust, and how the remaining animals respond as the system continues toward the deeper phases of ecological change that follow. When entire regions of the ocean fall quiet, the absence of movement becomes more than a local phenomenon. It is a sign of pressures building across scales larger than any single bay, estuary or basin. These low oxygen areas develop slowly, shaped by interactions between climate, circulation, nutrient inputs, and the responses of organisms that remain after the decline begins. As they grow, retreat, return, and reorganize, they reveal patterns that speak not only to the present condition of marine ecosystems, but to the direction those ecosystems are moving. These silent places do not tell a story of sudden collapse. Instead, they describe a trajectory shaped by cumulative stress, showing how marine environments respond to extended periods of strain and how they reorganize when pushed toward fundamental limits. One of the clearest messages comes from the consistent expansion of low oxygen waters in multiple parts of the world. Coastal dead zones linked to nutrient inputs have grown in frequency and size over the past several decades. At the same time, oxygen levels in the open ocean have decreased, forming mid-water layers where species once abundant now struggle to survive. These patterns reflect not only higher nutrient loads from land, but also increases in water temperature and changes in large-scale circulation. A warming ocean holds less oxygen and strengthens stratification, reducing mixing between layers. When these physical changes combine with biological factors, hypoxia spreads. The silent places along the seafloor and within the mid-water layers reveal how sensitive the ocean is to shifts in temperature and mixing. These quiet regions also demonstrate how small chemical changes can trigger broad ecological responses. The formation of a hypoxic zone starts with modest reductions in oxygen. When those reductions persist, organisms adapt by slowing, shrinking, or relocating. Over time, these adjustments alter the composition of the community. Benthic species that once processed organic matter may disappear, leaving microbes to dominate decomposition. Predators that once regulated certain populations retreat. The ecosystem becomes defined by organisms that tolerate low oxygen or exploit boundary zones. This shift in community structure shows that even moderate oxygen, declines, can produce changes that ripple upward through the food web. Another message found in these regions involves the relationship between nutrients, productivity, and oxygen demand. Over many years, nutrient inputs from land have increased dramatically due to agricultural runoff, wastewater discharge, and atmospheric deposition. These nutrients support large phytoplankton blooms. When the blooms die, they fuel microbial decomposition in the deeper layers. The microbes consume oxygen as they break down organic matter, creating conditions that generate or intensify hypoxia. Even when efforts reduce nutrient inputs, the internal cycling within the system may continue to support oxygen decline for years. These regions illustrate how long-term nutrient enrichment creates persistent pressures that require sustained reduction before recovery can begin. Many of these areas also show how quickly hypoxia can develop once the conditions align. In some coastal systems, hypoxia forms within weeks once stratification is established and organic matter begins sinking. The rapid onset contrasts with the slow pace of recovery. This imbalance reveals the vulnerability of marine ecosystems to sudden shifts and the lag that occurs when the system attempts to reverse course. The silent zones along the seafloor demonstrate this pattern repeatedly. They expand quickly when temperatures rise or when nutrient loads increase, yet they retreat slowly, requiring strong mixing or long periods of reduced input. Some of the most revealing insights come from regions where hypoxia cycles seasonally. These areas show how organisms adjust to repeated stress. Fish migrate away each year, returning when oxygen improves. Bivalves and worms slow their metabolism when oxygen drops and revive when mixing occurs in autumn or winter. Microbial communities shift between aerobic and anaerobic pathways depending on the oxygen level. These cycles reveal the dynamic interplay between physical forces and biological responses. They show that life continues even when conditions become difficult, but also that repeated exposure to stress changes the long-term structure of communities. In regions where hypoxia becomes more persistent, the changes intensify. Microbial processes dominate decomposition almost entirely. Chemical gradients become more entrenched. Compounds like hydrogen sulfide and ammonium accumulate in deeper layers. These shifts reveal how ecosystems reorganize when oxygen remains absent for long periods. They also show that recovery becomes more challenging once the system enters a stable low oxygen state. The sediments may release nutrients that sustain hypoxia even when surface inputs decline. The microbial communities may adjust so deeply to anaerobic conditions that reintroducing oxygen produces abrupt and sometimes damaging chemical shifts. These patterns show that once a system transitions to a low oxygen regime, reversing the trend requires substantial and sustained change. The movement of dead zones also offers clues about how changing circulation affects marine environments. Hypoxic waters that shift due to currents create sudden changes for organisms living in their path. Their appearance in new areas demonstrates the sensitivity of marine ecosystems to circulation patterns. When currents weaken due to climate-driven shifts, oxygen transport declines. When upwelling intensifies in certain regions, oxygen-poor deep water may reach the surface. These changes affect not only where hypoxia forms, but also how long it lasts. The silent zones reflect these broader shifts in ocean circulation and give insight into how future climate patterns may reshape marine habitats. One of the broader implications of expanding hypoxic zones is the compression of habitable space. As the lower layers lose oxygen and the upper layers warm, marine organisms become confined to narrower vertical bands. Fish move higher in the water column. Zooplankton restrict their migrations. Predators concentrate in limited depths where their prey also gather. This compression increases competition and alters predator-prey dynamics. Over time, it can reduce the diversity of species able to coexist. The silent places reveal how declining oxygen compresses habitats and forces organisms into crowded spaces where ecological relationships shift dramatically. These regions also show how complex feedback loops shape the ocean's response to environmental pressure. Hypoxia increases when microbial decomposition intensifies. Decomposition intensifies when more organic matter sinks. More organic matter sinks when nutrient inputs rise or when stratification traps productivity near the surface. Stratification strengthens when temperature increases or fresh water input rises. Each link in the chain reinforces the next. These feedback loops make hypoxia more persistent once it begins. They also show how interconnected the system is. Addressing any single factor, nutrients temperature mixing, affects the whole chain. The silent places demonstrate the interconnected nature of ocean chemistry, physics, and biology. Another insight from these zones is the role of resilience. Some ecosystems rebound quickly when oxygen returns. Fish re-enter the area. Benthic invertebrates recolonize the sediment. Microbial pathways shift. But other ecosystems struggle to recover. Once certain species disappear, they may not return even when oxygen improves. Sediments enriched with sulfide or ammonium may take years to re-equilibrate. These variations reveal that resilience depends on community structure, physical conditions, and long-term environmental trends. The silent places show where resilience remains strong and where it has weakened. The presence of these regions also highlights the importance of monitoring. Hypoxia cannot be understood through occasional observations. It requires continuous measurement of temperature, salinity, oxygen, nutrient concentrations, circulation, and ecological responses. Long-term datasets reveal patterns that short-term studies cannot capture. They show how hypoxia grows gradually, shifts suddenly, or retreats unpredictably. The silent places remind researchers of the need for sustained observation in order to understand the trends that shape marine environments. A deeper message found in these regions concerns how human activities influence the ocean. Nutrient runoff from agriculture, fossil fuel emissions that drive warming, and changes to land use patterns all contribute to the expansion of hypoxic zones. The silent areas below the surface capture the cumulative effects of these actions. Their growth and persistence demonstrate how closely connected the land and sea are. Even distant human activities affect the chemistry of the ocean. These connections reveal that addressing hypoxia requires more than local action. It demands coordinated efforts across watersheds, coastlines, and atmospheric systems. Yet these regions also remind us that the ocean has mechanisms for recovery. Mixing events, storms, and shifts in circulation can reintroduce oxygen. Reductions in nutrient inputs can weaken the feedback loops that sustain hypoxia. Changes in climate patterns can alter stratification. The potential for recovery exists, though the path may be long. The silent places reveal both the vulnerability and the resilience of marine ecosystems. They show that the ocean responds to pressure with measurable changes, but can also respond to improvements with equally significant adjustments. In areas where hypoxia persists but does not eliminate all life, the communities that remain offer examples of adaptation. Organisms shrink, slow, or modify their behavior. Microbial empires flourish under conditions hostile to other life forms. Drifters navigate boundary layers where oxygen shifts rapidly. These adaptations demonstrate how life adjusts to new limits. They also show how ecosystems reorganize around the pressures of low oxygen. These patterns reflect what may occur more broadly if oxygen declines across larger regions of the ocean. The silent places provide a preview of how marine life might adjust on a global scale if current trends continue. They also offer insight into how food webs change under extended stress. When predators retreat, prey species may increase temporarily, altering feeding dynamics. When certain grazers decline, microbial processes may dominate. The structure of the food web shifts, creating new interactions and eliminating others. Over time, these changes influence the movement of nutrients, energy, and biomass through the system. The quiet regions along the seafloor reveal how these shifts unfold and how they shape the broader ecosystem. In deeper contexts, mid-water oxygen minimum zones show how widespread oxygen decline affects species distributions. Some migratory fish avoid these zones entirely, altering their routes. Certain squid species change their depth preferences. The reorganization of these migration patterns affects fisheries, predators, and prey. The shifting boundaries of these zones also influence where carbon sinks into the deep ocean, affecting global carbon cycles. These deeper silent zones provide clues about how large-scale patterns may develop in the future. Another insight concerns the uneven distribution of hypoxia's effects. Some regions with strong circulation remain resistant to oxygen decline. Others, particularly semi-enclosed seas, experience severe and persistent hypoxia. The contrast between these regions shows how physical structure influences vulnerability. This has implications for future planning, as areas more sensitive to stratification and nutrient inputs will require more intensive management to prevent or mitigate hypoxia. These silent areas also highlight the importance of understanding the long-term consequences of chemical shifts. Decreases in oxygen affect not only the immediate habitat, but the cycling of nitrogen, sulfur, carbon, and metals. These chemical changes influence productivity in surface waters, the stability of sediments, and the balance of greenhouse gases. The patterns observed in hypoxic zones reveal how long-lasting these chemical shifts can be. They suggest that even if oxygen returns, the chemical footprint may persist for years, continuing to shape biological responses. The movement, expansion, and return of hypoxic regions also suggest that future oceans may become more fragmented. Instead of continuous habitats, the ocean could develop more isolated pockets of livable and unlivable areas. Species may become confined to narrow ranges, and migration routes may shift. These changes would alter the global distribution of marine life. The silent places show early signs of this fragmentation, revealing how sensitive species distributions are to oxygen levels. Despite these challenges, the presence of hypoxic zones also highlights opportunities for adaptation and management. Reducing nutrient inputs can stabilize conditions. Protecting habitats near the margins of hypoxic zones can preserve biodiversity. Monitoring circulation changes can help anticipate shifts. The insights gained from these regions can guide strategies designed to maintain ocean health. These silent places serve as a reminder that the ocean responds to both negative and positive influences. They show that future conditions depend not only on natural processes, but also on the choices made by human societies. As these quiet regions expand or retreat, move or return, their patterns offer a fuller picture of how ocean ecosystems respond to change. They illustrate the complex interactions between physical forces, chemical processes, and biological responses. They reveal the ongoing transformation of marine environments under pressure, and they prepare the way for understanding the next shifts that occur as the system continues evolving, shaping the conditions for the layers below, where deeper adjustments wait to unfold as oxygen becomes scarce, in ways once seen only in small, isolated pockets of the sea. When a region of the ocean has weathered cycles of oxygen loss, chemical shifts, altered food webs, moving boundaries, and long stretches of uneasy stillness, there comes a point where the water reaches a quieter state. It is not an end. It is a transition shaped by everything that happened before it. The seafloor that once held a full community of animals now supports slower, smaller survivors. The microbial layers that rose to prominence remain active, sorting and reorganizing the chemistry around them. The drifting organisms above continue moving through narrow bands of livable space, responding to faint cues in the water. Everything that persists within these waters does so because it has adapted to the limits imposed by the decline. When the deeper layers turn still, the transformation becomes easier to observe. It reflects a system that has adjusted as far as it can under the pressure, revealing the final steps in a long sequence of changes that began at the surface. In regions where hypoxia continues year after year, the seafloor transforms gradually. Sediments become more enriched with the remains of organisms that once populated the area. The breakdown of this material slows without oxygen, allowing layers of partially decomposed matter to accumulate. Microbial communities grow through these layers, converting compounds in ways that maintain a low-energy equilibrium. Certain types of worms burrow slowly through the sediment, reshaping small pockets but leaving most of the structure intact. Bivalves that once filtered water actively now sit deeper into the sediment, surviving through intermittent activity. Their shells show signs of chemical stress, etched by changes in pH and exposed to compounds that accumulate where oxygen does not reach. These organisms remain, not because the conditions favour them, but because they have found ways to make their reduced demands fit within the constraints of the environment. Above them, the boundary between oxygen-poor and oxygen-rich water may shift slightly with each season. When stratification strengthens, the boundary rises, compressing life into a thinner band. When storms or cooler temperatures mix the water, the boundary retreats. But over time, even these movements settle into a predictable pattern in long-standing hypoxic areas. The region above the boundary hosts animals that have adjusted their behaviour to predictable change. Zooplankton move cautiously, drifting at depths that balance feeding and survival. Small fish position themselves where they can avoid descending into dangerous depths while still reaching prey. The drifters that populate this band have learned the rhythms of this shifting layer. Their presence reflects the stability of a system that remains under stress but has found a balance within it. The deeper portion of the hypoxic region becomes increasingly shaped by chemical structure rather than biological activity. Microbial processes dominate, maintaining gradients through steady reactions involving nitrogen, sulfur, carbon, and metals. Nitrate becomes depleted. Ammonium accumulates. Hydrogen sulfide forms and is consumed. Methane emerges in deeper sediments. These pathways reflect a system that has reached a kind of long-term consistency under low oxygen. The interactions between microbes stabilize these processes, even though they create an environment where few larger animals can survive. This consistency is not a return to a healthier condition. It is a new baseline created under pressure. In some areas, water from these deeper layers occasionally rises due to changes in currents or slight disturbances. When this happens slowly, the chemical layers shift without much disruption. When it happens quickly, it can produce sudden changes in the mid-water column. Drifters respond by adjusting their positions, sometimes moving upward to avoid pockets of water that carry hydrogen sulfide or low pH. Because they have adapted to fluctuating conditions, their movements reflect a sensitivity to small changes in the water's chemistry. The narrow band where oxygen exists becomes a region of constant negotiation, shaped by each new disturbance. The animals that remain in these systems over long periods leave their mark. Their shells, skeletons, and organic material become part of the sediment structure. Over decades, sediment cores record the history of these transitions. Researchers examining these cores find evidence of community shifts, chemical changes, and periods of adjustment that trace the development of hypoxia. The sediments show when certain species disappeared, when microbial processes intensified, and when the system settled into its present structure. These records reveal a story of gradual transition rather than abrupt change. They show the long-term consequences of persistent low-oxygen conditions. For many years, these regions continue to operate in a pattern that reflects both stability and limitation. The animals at the boundary maintain populations proportional to the available oxygen. The microbial layers in deeper regions cycle compounds at steady rates. The sediments accumulate material slowly. Currents occasionally disturb the structure, but the system returns to its low oxygen state. This pattern persists until outside forces intervene, or until larger environmental shifts alter the system's foundation. One of the more subtle developments in these regions involves the presence of occasional visitors. Some species from better oxygenated waters drift into hypoxic regions when conditions near the boundary improve. They explore cautiously, feeding along the edges. When oxygen declines again, they retreat. These movements show how the boundaries between hypoxic and healthier waters shape the behaviour of mobile species. Over time, these species may adapt to the fluctuations, incorporating the edges of hypoxic regions into their regular movements. For them, the region becomes a transitional habitat rather than a barrier. The organisms that remain permanently within hypoxic zones do not rely on such movements. Their presence is tied to the long-term conditions. They survive within narrow limits and reproduce slowly. Over generations, their traits become tuned to the environment. Their reduced physiological demands reflect an alignment with the chemistry of the region. These organisms form the core of the stable community, anchoring the ecosystem even as the upper layers shift. Their persistence shows that even under significant stress, life finds pathways to continue. The presence of these long-standing hypoxic regions also affects the surrounding waters. The boundary between livable and unlivable water becomes part of the broader ecosystem, shaping migration patterns and influencing where predators and prey interact. This boundary can stretch for miles, creating a zone where certain species gather. These gatherings influence feeding, reproduction, and survival. As hypoxia persists, these boundaries become more defined, altering how the ecosystem functions around them. Some animals rely on these boundaries to avoid predators. In oxygen-rich waters, predators may follow them. In hypoxic waters, predators may retreat. The boundary becomes a refuge for some species, even though it represents a dangerous region for others. These dynamics show how hypoxia indirectly shapes behaviour, pushing species into new patterns that reflect the altered landscape. Another long-term development involves the chemical materials that build up in deeper layers. Compounds like ammonium, hydrogen sulfide, and iron-rich fluids accumulate over years. These compounds affect nutrient availability in subtle ways. When mixing occurs, they influence the chemistry of the water above, sometimes promoting productivity and sometimes suppressing it. These interactions create feedback loops that sustain the low oxygen state. Even when surface nutrient inputs decline, the internal chemistry of the system may maintain hypoxia for long periods. This persistence reflects the self-reinforcing nature of these regions. In deeper contexts, hypoxic and anoxic basins develop with little direct influence from human-driven nutrient loads. Their development is tied to circulation patterns, basin shape, and long-term climate conditions. In these areas, the transition toward hypoxia takes longer, but once established, the system remains stable. The deep layers become nearly devoid of oxygen. Microbial processes dominate. Chemical gradients become sharply defined. These basins show how long-term environmental shifts can produce persistent low oxygen conditions even without strong nutrient inputs from land. When examining these regions over decades or centuries, the larger marine system shows signs of adaptation. Species distribution shift, migration routes change, productivity patterns fluctuate. Some regions become hotspots for low oxygen survival strategies. Others remain resistant due to strong circulation or unique environmental features. The entire system adjusts in response to the growth of hypoxic regions. These adjustments show how interconnected the ocean is and how changes in one region influence others. Some of these adaptations involve changes in the structure of food webs. Predators that once relied on deep water prey may switch to species near the boundary. Zooplankton that migrated across many layers may restrict their movements. The simplification of food webs in hypoxic zones also influences adjacent areas. When fewer species persist within hypoxic regions, the areas around them may experience altered patterns of feeding, grazing, and predation. These shifts occur gradually, shaping the overall dynamics of the ecosystem. Over time, the long-standing hypoxic areas become integrated into the broader mosaic of marine habitats. They remain distinct but influence the movement of nutrients, carbon, and organisms. Their presence affects where certain species breed, feed, or take refuge. In some areas, the boundaries between hypoxic and healthier waters shift slightly with climate-driven changes. Warmer years push the boundary upward, cooler years allow it to sink. These subtle shifts reflect the ongoing influence of the broader climate system. The influence of these regions extends far beyond the immediate area. When hypoxic water reaches areas with strong mixing or upwelling, the chemistry it carries affects productivity. If ammonium reaches surface waters, phytoplankton may respond rapidly. If hydrogen sulfide or other reduced compounds rise, the response may be slower or more controlled. These chemical exchanges connect hypoxic regions to healthier waters, shaping the productivity of nearby ecosystems. Even as these regions remain under stress, the organisms that live there continue adjusting. They respond to slight improvements by increasing their activity. They respond to declines by slowing again. This responsiveness shows the flexibility of life, even under difficult conditions. The slow pace of these adjustments resembles a steady transition rather than a constant crisis. The ecosystem continues shifting, but it does so in a way that follows the pressures imposed on it. In long-standing hypoxic regions, the relationship between physical forces and biological responses becomes even more evident. Seasonal changes continue to influence the boundary layers. Currents redistribute water masses. Occasional mixing pushes oxygen downward. These forces create small variations in the structure of the region. The organisms that remain adjust to these subtle shifts, maintaining their presence within narrow limits. Some areas experience slow, long-term recovery. When nutrient inputs decline, stratification weakens, or circulation patterns shift, oxygen may gradually return to deeper layers. The return is rarely immediate. The system must adjust chemically first. Microbial processes must change. Sediments must re-equilibrate. Over time, the deeper layers shift from anaerobic to microaerobic and eventually to oxygen-rich conditions. When this happens, organisms begin returning cautiously. Worms recolonize the sediment. Bivalves settle in new patches. Fish explore the area. These early signs of recovery reflect the ongoing potential of the ecosystem to adjust under improved conditions. Recovery does not erase the history of hypoxia. The sediments retain the chemical imprint. The structure of the microbial community remains shaped by years of low oxygen. The physical boundaries of the region may stay defined even as oxygen returns. But recovery changes the direction of the system. It allows life to expand outward from the narrow bands where it survived. It allows the ecosystem to build new pathways for energy and nutrient flow. The regions that show partial recovery provide important insight into how much resilience exists within marine ecosystems. They reveal that even after years of stress, systems can adjust if conditions change. The pace may be slow, and the outcome may remain uncertain, but the potential remains. These areas highlight the flexibility of life and the adaptability of chemical systems. At the edges of long-standing hypoxic zones, transitions between quiet and active waters reveal the complexity of marine resilience. These regions illustrate the gradient between stress and recovery. They show zones where life finds footholds and where new patterns emerge. They become areas of continued study for understanding how ecosystems reorganize under pressure and how they respond to change. The processes unfolding in these silent regions show a story written over long periods. It is not a story of endings, but of transitions. It reflects shifts in chemical pathways, biological adjustments, and the interactions between physical forces and ecological responses. These regions continue evolving, shaped by the pressures around them and by the organisms that persist within them. They remain quiet but active, limited but alive, reduced yet functioning. Even as they reveal the pressures placed on the ocean, they also show the persistence of life. They remind us that ecosystems continue adjusting, as long as conditions allow it. They demonstrate that change in the ocean does not happen all at once, but through slow reorganization. And they set the stage for ongoing shifts that continue beyond any single region, marking the beginning of further transitions that emerge as the larger ocean moves toward new conditions shaped by the forces acting upon it.